Welcome back, everybody. We made it to day 3. I'm glad you're still with us. Glad you're here with us today. A lot to do yet today. Pretty exciting agenda, I think. So first off, again, I want to extend a big thank you to our sponsors making this event happen. So again, please, if you haven't already, go to the lobby, go to the exhibit hall, visit the sponsors, see all the great stuff that they've got available, chat with them, Zoom with them if you can. We're very grateful to them for supporting this effort and this event. A couple business items as well. One little detail, for those of you whose browsers are not auto-refreshing for the chat box, you can put your cursor over the blue box, right-click, and click Reload Frame for chat. And what that will do is reload the frame without reloading the whole page, so your video stream won't be interrupted. Secondly, please click on the conference survey and take that survey. Again, you can keep it open and rate presentations as you go through or make comments. But we'd really love to hear from you, get your feedback on how things go. And just a little side, I'm a bit of an extrovert, so it's really hard to gauge how everybody is feeling and thinking and taking in this stuff. So the conference survey is even extra important for us to get that feedback from you. Also, downloads, please click the downloads if you're interested in some of our material, particularly the advocacy toolkit. And I'll give a quick little plug. Our Jake and Jason are working really hard right now with our advocates in Wisconsin. We are actually very close to getting a bill introduced for a $3 million appropriation for research. We've got weeks to get that done. We would love to get your support in emailing, writing, calling legislators, actually right now for the next week or two. Really important. We're grateful to the Christopher and Dana Reeve Foundation for writing a letter of support that we passed off to legislators. So if you represent an organization, we would also love to get a letter of support from you. You can reach out, you find my email on the website, or Ryan or Lucia, who you've been corresponding with. One more thing. Email contact info. You'll get an email this week from us asking about your willingness to put your contact info out for all the attendees so that you all can connect with each other as you like. Okay, so let's move on a little bit. Also, one other thing, sorry. If you have any questions about any of the stuff in the agenda, any of the presentations, any of the panel discussions, any questions that didn't get answered or asked, or maybe even didn't understand some of them, feel free to reach out to us. Reach out to me. Happy to talk with you, happy to engage with you on some of that stuff. And remember, for all attendees, there will be an available link to watch everything, so you can watch it again long before it goes up to YouTube. And so the last couple of days, you have heard a number of presentations. So as I said, the first day was a lot of contextualizing stuff, a lot of background, a lot of the complexity beyond the science and the intersection of advocacy and science. And then yesterday, heard some clinical innovations, some sexual function research. In the clinical innovations, almost a kind of message of sometimes there are things learned and understood or hypothesized that challenge the dogma. And it's really important for us to be aware of that stuff, knowledgeable about it, and in some cases, ready to advocate on behalf of some of those new understandings and new learnings. Sexual function research, similarly, is an example of, this is an area, there aren't many people doing it. It's complicated stuff. As you heard from some of the presenters, sometimes difficult to get funded and even understood. And it's very important for us as advocates, if that's a meaningful, it is meaningful, but to the degree that, we folks with SCI, as a parent with a son, we want everything, right? So there's a time also to raise our voices, and it also, from the presenters you heard, there's been a little shift in that, and there's been more attention paid and some more funding available to do that work. And as in many ways, that's as a result of some people raising their voices about that. So today we're going to hear from 3 presenters on some regeneration strategies. And then we're going to have a panel discussion with them. I think that'll be very exciting. And then the rest of the day will be spent on spinal stimulation, and in a whole range of different perspectives and different approaches. And then the exciting news towards the end of the day of ONWARD, company that's bringing a product to market, to test and to market. So let's get into the program. Look forward to talking with you. Please welcome Michael Lane, Associate Professor of Neurobiology and Anatomy at Drexel University, College of Medicine. He will be talking about stem cells, but particularly neuronal cells, and particularly spinal interneurons, and some of the research he's been doing in cervical spinal cord injury and some of the conclusions he's come to. Welcome.
I want to thank Unite to Fight Paralysis for giving me the chance to speak with you today. I'm going to be telling you a little bit about our work at Drexel University in the Marion Murray Spinal Cord Research Center, where we're looking at transplanting neural precursor cells, or developing building blocks of the spinal cord, to repair the injured cervical spinal cord. I firstly want to thank many of the collaborators that I have at Drexel University and within the Marion Murray Spinal Cord Research Center, as well as the external collaborators that work with me around the US and overseas on this project and other related projects. I want to also thank the financial support that we receive from foundations and government institutions, which supports this work and other related work as well. I need to also thank my research team, which comprises graduate students, medical students, postdocs, undergraduate students, and even high school students who all volunteer their time to work on these projects and essentially contribute in very important and meaningful ways to the development of new ideas, new research projects, and those that have contributed to the work that I'm talking about today, I'll highlight as I move through some of the results that I present. Lastly, I need to thank Jake Chalfin, who's an individual living with cervical spinal cord injury, who volunteers to work with us as a consultant, listens to our ongoing discussions about our results, talks to us about our future plans, and also reviews some of our proposals that we submit for grant applications. As I said, our research team is interested in transplantation for promoting spinal cord repair, but our focus is on cervical level spinal cord injury. Most of you will know that the majority of injuries occur at cervical levels. In the US, that number is about 60%, and overseas it's even greater. But with these injuries, the vast majority of injuries actually also have some degree of anatomical sparing, and that sparing of tissue serves as a substrate which can facilitate plasticity. So plasticity is where the spared anatomy can be tapped into to even spontaneously start becoming more active or even form new connections through which it can actually enhance the degree of recovery or functional improvement, which is functional plasticity. So we're interested in studying how this actually occurs after cervical spinal injury, specifically with how it affects respiratory networks. So after a cervical level injury, there's a huge range of different deficits that can occur, among which is impaired breathing. So impaired breathing actually is a key contributor to morbidity and mortality in individuals with cervical level injuries. And even those people who don't require assisted ventilation, there may be ongoing deficits and even things that go unnoticed as sleep disorder breathing that can contribute to impaired quality of life, and that may even become life-threatening with time. So what we're looking at here is a schematic diagram of the neck of an adult male and with an overlay of the cervical spinal cord in this image here on the left. On the right-hand side, we have a schematic diagram showing you what an MRI side-on would look like, highlighting the brain and spinal cord. And this highlighted area here is showing a degree of vertebral disruption, compression of the spinal cord, and the spinal cord injury. Underneath that, we have an actual MRI from a patient where the vertebral column you can see is compressing on the spinal cord, resulting in contusion, hemorrhage, and edema. And in this red inset image, we have a cross-sectional view of this same injured spinal cord. So we're actually not studying spinal cord injury in patient population. We're actually looking at it from a preclinical standpoint. So we're using an animal model to reverse translate what we learned from the clinic and recapitulate that in animal models to better understand how these injuries affect respiratory networks and what potential there is for plasticity and more importantly, how we can therapeutically harness that with neural transplantation. So here's a schematic diagram of the adult rat, which is the primary model that we use. Again, with an overlay of the cervical spinal cord. And when we look at the structures within this cervical spinal cord shown here and color-coded, we can start to tease apart individual neural networks. Specifically here, we're looking at the phrenic motor network, and the phrenic motor network innervates the diaphragm, which is a primary muscle of respiration. As I said, this is color-coded here to show you the individual components. So in green, we have the phrenic motor, spinal phrenic motor system. So you have phrenic motor neurons, which innervate the muscle directly. We have spinal interneurons, which innervate and regulate these motor neurons. So these spinal interneurons can be inhibitory. They can actually suppress motor output, or they can be excitatory and excite the degree of motor activity, or they can even be modulatory. So modulatory cells can increase or decrease the gain, if you like, of motor function. And in doing so, they can increase or decrease amplitude of activity. So what you're looking at in blue is descending input from the brainstem or brain, which provides respiratory control to this spinal network, activating the network itself and resulting eventually in diaphragm contraction. But following an injury, as shown here in red, is a contusive type injury, which is the model we use preclinically. What essentially results from this injury is a loss and damage directly to the phrenic network itself. So you're losing these components of the network, and you're compromising these descending axons. So you're losing input from these respiratory centers in the brainstem. So in order to look at the functional deficits, we can stick wires directly into the diaphragm itself and record activity or record muscle contraction. And that's what these green traces are showing you here. So on the side of this injury, which is a one-sided injury, this side is showing you decreased muscle contraction compared to the intact side, the spared side. This ipsilateral diaphragm has reduced muscle contraction during each inspiratory event, which is what you see here. That each one of these contractions is an inspiratory event or muscle contracting, which inflates the lungs. In between, obviously, where there's no contraction of the diaphragm, we have expiration, the passive event. And as you can see, the contralateral side keeps working quite well. We can drive the system harder and present a challenge to these animals by either decreasing oxygen or increasing carbon dioxide. And that makes the respiration increase and makes the muscles contract more. So by driving the system harder in this fashion, providing a respiratory challenge, we can stress the system and see how it responds. And what we're learning is that following these injuries, the system, the injured network cannot respond in the appropriate way. So you want it to increase muscle contraction and it can't, or at least not that much. This is a form of respiratory insufficiency seen both in people with these injuries and our animal models. So as I mentioned, we have an interest in these spinal interneurons and how they can represent therapeutic targets. Interestingly, work by our research team and the work of others looking at locomotion and non-respiratory functions, spinal interneurons have amazing capacity to form new networks. So even after their original connections might be lost or damaged by an injury, they can use this residual tissue to sprout new axons and form new connections, providing essentially a new anatomy after spinal cord injury. And this new anatomy, recruiting these spinal interneurons, can contribute to plasticity that can enhance the degree of recovery that we see. So what sort of interneurons exist? To do that, we can look at the actual spinal cord itself. And this is a section taken through the adult rat spinal cord, and we've stained it for individual components. So in red, we've stained for neurons. And as you can see, they fill the middle of the spinal cord and create this butterfly-like pattern, surrounded by this green area, which is actually descending axons from the brainstem and brain. In blue, we've stained for a subset of neurons, and these are cholinergic neurons. And so these big neurons here are actually motor neurons, which are cholinergic, which send their axons out to the muscles and help the muscles contract. There's also a subset of spinal interneurons, which are also cholinergic, and these are modulatory spinal interneurons. So as I said before, modulatory neurons can increase or decrease the gain, if you like, of motor activity. So that's actually quite interesting here. After a spinal cord injury, where we need to increase the amplitude of muscle contraction, maybe these cells represent an ideal therapeutic target. So we firmly believe, along with others, that spinal interneurons are key components of plasticity after spinal cord injury that could be therapeutically harnessed. The question, of course, is which interneurons do we need to be looking at? So we've identified two cells within our phrenic network that exist within the phrenic network normally. So these cholinergic modulatory cells, known as V0C, and also a population of excitatory cells that can increase motor activity, the V2As. Both of these cells exist within the phrenic network normally, And interestingly, after a spinal cord injury, they increase their connectivity. So they're already contributing to anatomical plasticity and forming a new anatomy. Likewise, the cholinergic cells also contribute to increased connectivity spontaneously after injury. And interestingly here, if we pair a rehabilitative strategy like an exercise-based therapy or activity-based therapy like respiratory training, we can increase the number of these cholinergic cells that also connect. So we can therapeutically drive that connectivity. The question, of course, relative to today's talk is, can we harness these populations of spinal interneurons for transplantation? As I said, our two areas of therapeutic strategies that we're studying in my research team are activity-based therapies or rehabilitation. And in order to achieve repair, we're using this cell-based strategy of transplantation of neuroprogenitors. The schematic diagram is going to walk us through essentially how neuroprogenitor cells can be used to repair this damaged, disconnected spinal cord. So we have a big injury site here in the middle of the spinal cord shown here in gray, and we can transplant donor cells, our neural precursor cells shown here in green, and we can actually literally label our cells that are green so we can track where they are. And when we've done this, much like other people have shown, these cells survive in the injury site, they restore tissue continuity, where there would otherwise be a gap or a loss of tissue, and they provide the building blocks for repair, these developing neurons and glial cells. We can also start to see where these developing neurons extend their axons. Are they connecting to the injured host spinal cord? Specifically, in our case, do they connect to the injured phrenic system? And we can label our phrenic motor system with red dye, as shown here. And these red labeled cells are actually receiving inputs from our green donor neurons. So they do get inputs or form a new network with our injured phrenic system. We can also start to see which sorts of cells reside within our transplant, and certainly subsets of these cells, as we might hope, are cholinergic interneurons. So we have the right types of cells, at least partially. We can also start to look at descending input. Does the host or the injured axons that have been damaged by the spinal cord injury, can they make connections with our donor cells? And it turns out they in fact do. They extend axons into the transplant and form connections. So essentially we've established an anatomical relay between our injured axons from the host connecting to our donor cells. The donor cells then connect to our damaged phrenic network, and that can restore an anatomical path through which we can restore functional recovery. We can even start to see how other axons might feed into this transplant network to regulate its activity. But essentially what we've shown, much like the work of others transplanting neural precursor cells, is that they can integrate with the injured spinal cord and they can improve functional outcome. Unfortunately, the degree of functional recovery is incomplete and the connectivity that they can achieve is inconsistent. So these are two things we now want to address. Essentially, can we better refine the populations of cells we're using, so the types of neurons or phenotype of neurons, and can we improve their connectivity? So addressing the first question first, and that's about donor cell phenotype. What sorts of cells exist within our neural precursor cells? Some research done by one of my graduate students a few years ago in collaboration with a group at University of Texas, led by Shelly Sakayama, Albert. We looked at the donor cells we were transplanting, and essentially using these genetic markers, we can track what sorts of cells we have. And unfortunately, our cholinergic cells and our excitatory neurons are being lost within our transplant donor tissue. And we're left with these inhibitory cells, which inhibit motor function and might be suboptimal for promoting recovery. So what we did was work with Shelly Sakayama's team to start engineering from stem cells new populations of spinal interneurons. And when we look at the results from this, we look at muscle activity. In those animals that received no transplant, as you might imagine, muscle activity was significantly reduced. With the transplant, we could improve that, and that's just with this mix of different neuronal precursor cells. But when we enrich this for specific populations of interneurons, like these excitatory V2A, we further enhance the degree of recovery. We can also actually modify these donor V2A cells to see how they contribute to recovery, which is what we're now doing. And we can modify them so that we can apply drugs to the spinal cord and turn off the donor cells. And that's a way of confirming here that the donor cells, when we turn them off, have been contributing to this muscle contraction. So they are actually promoting this recovery that we see. Of course, we actually only have looked at a few types of interneurons so far. We want to start expanding this to look at other types of spinal interneurons, and we want to preserve and promote connectivity in a more consistent fashion. To do that, we combine our cell therapy with activity-based training or rehabilitation. So this is tapping in once more to our respiratory training strategy, where we lower the oxygen that animals are breathing after they've received a transplant. This increases activity within respiratory networks, but also strengthens the connectivity of our donor cells to our injured frank network. And in fact, you see here from the results, the number of cells that increase at connectivity are really significant. The question, of course, is which interneurons are connecting, right? So when we look at our donor cells, once again, we do have some populations of cholinergic interneurons, but not many. So again, we need to increase the number of relevant populations of interneurons within these transplants. So in summary, we know that spinal interneurons can mediate plasticity after injury. We know that connectivity can be enhanced with strategies like activity-based therapy and training. We know that transplanting spinal interneurons improves outcome after these types of cervical level injury. And by pairing the two together, transplantation and activity-based therapy, we can strengthen connectivity of the donor cells with our transplant. The question now, of course, is what other types of interneurons can we look at transplanting into the injured cord? What other types of combination therapies could be used aside from activity-based therapies? Can we use intraspinal stimulation or epidural stimulation, for instance? And through collaboration and discussion with other researchers and also consultation with Jake Chalfin, we're interested in looking at moving into more chronically injured spinal cord, which is obviously more relevant to the larger population of people who are currently living with spinal cord injury. Well, we want these treatments to be developed and relevant for this larger population of individuals. And finally, we're looking at whether we can use non-neural cells as well, which might also be pro-reparative after spinal cord injury. So very briefly, I want to thank everybody that contributed to this work. My graduate students and postdocs, including Lana Jolu-Diva, who's now a postdoc at Gladstone Institutes, the other faculty members here at Drexel University who've assisted me with this work, and my collaborators at other institutions within the US and overseas. And with that, I'll take any questions with any time we have remaining. And thank you again for giving me the chance to tell you about my work.
Please welcome Kobe Koffler, a PhD and assistant professor of neuroscience at University of California, San Diego. Kobe will be talking about scaffolds in the Tuszynski lab and some of the research that they have been doing, looking at architecture to help reorganize the cord and the anatomy post-injury. Welcome.
Good morning, everybody. I'd like to thank the organizers for inviting me today to speak at Unite to Fight Paralysis meeting. My name is Kobe Koffler. I'm an assistant professor at the Department of Neurosciences, School of Medicine, UC San Diego. And I'm here today to speak about the work we're doing with 3D printed scaffolds for spinal cord injury repair. The challenge that we have with spinal cord injury is the breakdown of the spinal cord, which result in disruption in communication, disruption in the flow of information between the brain and peripheral organs. As you can see here in the schematics, we have bones that are pressing on the spinal cord, which is the white element that's in the center of this figure, and we have an injury depicted here in red. This injury also results in a breakdown of myelin, which is the component that encases the axons. Axons are the projections of the nerves, of the neurons that sits in the brain, and then they go down the spinal cord and out to the periphery. There is also secondary damage caused by the hostile environment that's within the injury site, which is toxic to cells. Everything breaks down. There's a lot of inflammation, and that increases the damage due to the injury and the compression from the bones, as you can see here in the image. All of that leads to a loss of motor and sensory functions. Now, the way the spinal cord is organized, we generally have two elements. We have the white matter in the periphery and gray matter in the center, and they are organized very differently. In the white matter, the axon, again, those are the projections of the neurons that come down from the brain. The descending axons are organized very nicely in parallel. They are linear, and they're packed together. And you can see this in the image here. This is general labeling for axons, and if the head is on the left and the legs are on the right, you can see how it's all nicely organized. While in the gray matter, there is disorganization. The axons are not organized at all, and they're randomly oriented in all directions. And so when we come to design experimental approaches to promote regeneration, we have a problem, and the problem is disorganization of regeneration. And here are some examples. When we look at host regenerating systems that enter the injury site after an implant, some kind of an experiment, we see distinct disorganization. You can see here several images. On the left, we have labeling, general labeling for axons. It's called NF200. In the center, we have labeling for a specific subsystem of axons that's called 5-HT, serotonergic axons. Those are axons that modulate motor movements. And then on the right, we have labeling for CST, corticospinal tract. And the bottom image here shows you what it looks like, what those axons look like within the injury site that is depicted here in white interrupted line. And in all images, you can see that the axons have no directionality and they're basically randomly oriented in all directions. Same is true for stem cells. If we inject stem cells into the lesion site, we get the same problem. You can see here a stem cell that extends processes, and these processes are again randomly oriented in all directions. There is no directionality, nothing. And so our approach for spinal cord injury repair is to try and reorganize regeneration using biomimicry design and principles. And the goal is to rebuild the white matter architecture, as I showed you before, in order to guide those regenerating axons down the spinal cord into their respective tract. The way we do that is we use linear microchannels that physically restrict and guide regeneration. So when an axon gets into the lesion site, there's only one way, and that's the other way. Here's what it looks like. We have an implant that contains many linear channels, and so when an axon, that here would be a green line, comes from the intact injury that's here on the right side, it would get into the injury site, see the microchannel, and then can go only to the other side, which is the intact spinal cord. Can't veer sideways, up, down, or backwards. When it gets into the other side, it's already in the same orientation and the same coordinates in space, and so we improve the fidelity of regeneration. The way we fabricate and manufacture these implants, we use 3D printing technology. Usually people think about inkjet 3D printers, where one would push material through a nozzle, and this is a different way. We use light. And so what we do, we take on the left side, you can see a section from a real rat spinal cord. We digitize that, make it a digital mask, and we'll load it to the printer. That mask is then being projected onto a pool of material that sits here on the stage, and using the light, the material is being polymerized, and we're building the structure in 3D. And here on the right side, you can see an example of an implant that contains linear microchannels around the white matter area, and we have a block of material where we have the gray matter. It provides stability from the core of the implant. Each channel is 200 micron wide, and this specific implant was designed for a rat experiment and is 2 millimeter long. We can also load stem cells into the scaffold. We can load stem cells into the white matter area where we have these open channels, and if we load neural stem cells, we are then creating something that we called a neuronal relay between the host, the stem cells, and the host intact tissue again. As you can see here in the image, on the left side we have several, in this case 3 subsystems of axons in different colors that are coming from the intact cord. They get into the injury site, into the scaffold with the linear channels, where they would meet stem cells, neural stem cells that are depicted in green, and those neural stem cells will send their own axons outside the injury site, outside of the channels, into the caudal spinal cord, as you can see here. And then again, each system is still oriented on the same pathway, the same coordinates in space, even stem cells, as stem cells as well as host systems. Another advantage of using 3D printing is the ability to do precision medicine. We can create scaffolds that fit precisely each injury. As we know, the spinal cord injuries are different between patients. And here in an example, we have two images of two different injuries, two different MRIs. We trace them in red. You can see how different they are. We can create a 3D model, print a different scaffold, and then fit that onto the MRI. So you can see how it fits precisely to the MRI as we acquire it. And so we decided to test those 3D printed scaffolds in rat studies, feasibility studies. The experiment is a T3 level of complete transection. We make a complete gap, complete cut with a gap in the rat spinal cords. We perform an acute implant, which is important. At the time of injury, we make the implant, and this is the scaffold that we implanted. Here is an image, just general understanding of what we do in terms of the surgery. In white here, you can see the rat spinal cord. There are some blood vessels that supply the spinal cord. We then create the transection. You can see here there is a gap, and then we implant. Here is the scaffold that was implanted. The two black arrows shows you the interface with the intact spinal cord. First thing we're looking at is stability. So you can see that the spinal cord, the implant of the biomimetic rat spinal cord scaffold, it's stable. 4 weeks after implant, the structure is still there, the channels are still there, the core is still there, and all of the channels are full with axons. They're labeled with green, and you can see that every channel has a field of axons. Next, we looked at stability after 6 months, long-term experiment. And as you can see on the left side, on the left image, the channels are still there intact. Mechanically, they're still there and stable, and they are filled with green stem cells. And this is important because remember, this was an acute implant. You can see on the right side on images A1 and A2 what happens when you perform an acute implant of just stem cells without the scaffold. You can see that there is poor cell survival and huge, very big cavities, those black holes that you can see on the images. And so not only that we have long-term, 6 months after implant, we have stable scaffolds, they are full with stem cells, but they also provide a protective environment for the stem cells from that hostile environment that we talked about at the beginning, right? So the stem cells themselves would not survive this kind of an implant. We next looked at something that we call the reactive cellular. Reactive cellular, anything that we implant into the in the human body would get some kind of a reaction. And so here's an example of what it looks like when we implant a scaffold from a material that we worked with before many years ago. It's called agarose. You can see in purple the extent of the reaction, the thickness of the reaction of what the tissue looks like. On the right side is the scaffold itself, the implant, and black is the interface with the intact spinal cord, and you can see the thickness of this purple tissue. On the right side, you have the same implant, different materials. This is the 3D printed scaffold from materials that called polyethylene glycol and gelatin, and you can see again the scaffold is on the right, and you can see how much smaller that reactive cell layer is, and not only it's smaller, it's less dense, and that's important. So we quantify that to be 35% less dense and less thick than what you see with previous materials. When we look at guidance, again, we talked about this before, what we were able to see inside those linear channels, the guidance and restriction of growth of regeneration. We can see that the axons are again nicely packed together and linear, if left is head and right is legs. And on the right side, you can see this previous image that I showed you. This is what happens when you put a stem cell into a lesion site without scaffolds, without linear channels. Axons are randomly oriented in all directions. We then looked at functional recovery. We performed two different assays. The first one is called BBB, free walking behavior, let the animal walk, and we observe how it moves its limbs. Now, the effect of the complete transection is that both hind limbs are completely paralyzed, flaccid, not doing anything. And you can see that animals that were implanted with empty scaffolds or with stem cell implant without scaffolds showed no recovery. No recovery. Again, remember, the stem cells didn't survive, so it's not a surprise. No recovery. While animals that were implanted with scaffolds loaded with neural stem cells showed improved recovery up to a score of 7. Score of 7 means that they're moving 3 joints independently. They're moving their heels, their knee, and their hip independently. This is still not full recovery. A full recovery would be 21. This is the beginning of recovery, but it's important because it's significant. The second type of test that we did was called electrical stimulation. We provided electrical stimulation in the brain and recorded signals from the gastrocnemius muscles in the leg. We just probed the leg for EMG signals. And what we found was that with animals implanted with scaffolds loaded with stem cells, we were able to record signals. So there was connectivity between the brain through the spinal cord into the peripheral organs, into the muscles in the legs. When we cut this connectivity above the injury site, we lose this signal. And when we look at animals implanted with scaffolds, empty scaffolds, no stem cells, there is no signals, no connectivity. And so to summarize what I showed you today, it is feasible to use 3D printed scaffolds to create biomimetic devices for spinal cord injury repair. The scaffolds are safe to implant. They are stable in vivo long term, which is important. They don't break down, they stay there to guide those regenerating axons. They support neural stem cell survival and provide protective environment. They guide host regeneration, and they support regain of function in the most severe model of spinal cord injury, which is the complete spinal cord transection. We are now in the process of further preclinical studies to translate and scale up the technology in order to hopefully bring it to the patients in the future. With that, I'd like to conclude. Thank you for listening, and of course I'd like to acknowledge everybody who contributed to the work on this project.
Now welcome Murray Blackmore, PhD and Associate Professor of Biological Sciences at Marquette University, who will be talking about gene therapy as yet another solution for regeneration. And of course, as with all of these, please be thinking about the intersection of the 3 presentations. Welcome, Dr. Blackmore.
Well, hello. I'm Murray Blackmore. I'm a professor at Marquette University, and I'll be talking today about gene therapy approaches to spinal cord injury. It's really an honor to be asked to present at this meeting. It's one of the most inspiring, but also one of the most intimidating venues. There's probably no audience that cares more about the progress that either has or hasn't been made. Let me start by thanking the many people in lab who have done all this work, the funding that we've received, There's a couple of great collaborators whose work I'll be highlighting, and especially Nancy Nicholas, who acts as our SCI community liaison. And her job is to attend our lab meetings each week and to provide that perspective, the lived experience, which I think is really important even at the level of basic research. Very briefly about me, I do come at this work from a couple perspectives. On the one hand, I'm this guy. I'm a scientist who enjoys puzzles, likes figuring things out. I'm also this guy with his mom. She was a C4 quad. This picture was taken about a week before she died. I took her home to see her garden one last time. And that was about 27 years after she was first injured, and it's been 8 more years since then. And that leaves me a lot of the time as this guy in the middle, trying to figure out how to make progress and trying to understand why has progress been so slow. There are a couple of things you need to know to understand the approach that I'm taking. It starts here in the embryo. That's where most of the neurons in the brain and the spinal cord are first made. They're initially just a cell body. That would be this little dot here signifying the cell body. It's where the DNA is located. It's where most of the cellular material gets made. Once that cell body is created, it does something remarkable. It grows this long, long extension called an axon. So that axon will extend a great distance. It will wire up to a target cell, which will then send its axon out into the body to carry some function. So it's a relay circuit. 2 things about these axons. One is the incredible length of the axon compared to the cell body. This is not to scale. The axon is actually thousands of times longer than the diameter of the cell, such that the great majority of the cell's material ultimately will be out in the axon. So it's this incredible feat of cellular construction. The other thing is the speed of the axon. A growing axon in an embryo can cover about 1 to 2 millimeters of tissue per day. All right, so these circuits get set up very early in embryogenesis, and then in the adult, they get used for function, and they can be somewhat refined, but they're largely stable. Now, there are millions of axons that travel in the human spinal cord. They travel both up and down the cord. They're coming from neurons, from these cell bodies, that are distributed all through the brain and all through the spinal cord. So many different sites of origin. These axons, they travel in different parts of the spinal cord. That's shown here. You know, some are in the front, some are in the back, some are anterior, some are more lateral, and so on. Which means that for any given injury, just based on the geometry of the injury, some types of axons in each person will be more or less affected. And of course, these different types of axons running in the spinal cord, they're all carrying many, many different types of functions. Some are for movement, some are for balance, some are for sensation, some are for bladder control, some are for temperature control, and on and on and on. Some obvious implications of all that. First, an injury to the spinal cord is going to disrupt a lot of functions for the individual. And I don't think I really need to explain that more to this audience, certainly. The other is that because injuries are often incomplete, leaving some axons spared, in some ways, each individual injury is going to be unique, right? It's going to leave each person with a slightly different set of functions that are disrupted or maintained. And again, I don't think I need to belabor that point either. But I do really want to acknowledge how important it has been to have someone like Nancy in the room to keep these things kind of clear to us. What these different types of axons have in common, though, is that if they are disrupted, if they're cut by an injury, they mostly lack the ability to regrow or regenerate back across that injury site. And that's the basic problem that we're trying to solve. What that means is that unless you can find a way to make that axon regrow or somehow reroute the information around the injury, there's going to be a permanent disruption of communication, which means a permanent loss of all those functions that I just talked about. Okay, so why can't these axons regenerate? Well, there's been a lot of work done, and I'm going to say there's sort of three main reasons that break into two categories. The first is here at the site of injury. There are particular cell types that act in the tissue to block the advance of axons. And they do that in sort of two separate ways. One, they produce these molecular cues that signal to the axons to stop advancing. Second, axons need a particular surface on which to grow. And at the injury site, that surface isn't available. So you might say the bridge is out. Those are often lumped together as extrinsic barriers to axon growth. They're coming from outside of the axon. Now, alongside that, the neurons themselves basically fail to reinitiate that growth program that they used in the embryo to first build the axon. This is often called the intrinsic barrier to axon growth. You can think of it as a car with no engine. And so obviously there, the idea would be to try to ramp up the intrinsic cellular growth machinery of the neuron to try to restore axon growth ability. The reason I like this metaphor is it makes it pretty clear that if you want to get axons to regrow across the site of injury, you're probably going to have to do three things at the same time. You're going to have to get rid of these molecular stop cues. You're going to have to provide some substrate for the axons to grow on, and you're going to have to rebuild that growth machinery that the neurons use to build axons in the first place. Today I'll focus more on that intrinsic side of the problem. So the question is, how do you rebuild this cellular engine for growth? And the problem with engines, of course, is that they're very complicated. So in order to advance an axon, a neuron has to engage a whole host of cellular processes. And each one of those processes, you can kind of think of it as the net output of the interactions of dozens to hundreds of gene products. So it's this really complicated system for growth. That's great if you're a basic scientist. You can make a whole career out of studying this. It's a problem if you just want the axon to grow. But remember, neurons do this embryonically. So how do they organize it? How do they accomplish it? And that's where these proteins called transcription factors come in. So that's another term you'll need to hang on to, transcription factor, or TF. I'll just call them TFs. These are proteins that work back in the cell body of the neuron. And what they do is they interact with DNA, and they make decisions about what kinds and what amounts of proteins that cell's going to produce. So the idea is that underlying this complexity of hundreds of interacting proteins is a much smaller set of master regulator genes, these TFs, that are basically orchestrating and controlling the process. So maybe that's the tractable therapeutic target. Maybe if we can deploy the transcription factors that organize axon growth, we can start to rebuild the growth capacity in the adult neurons. That's the basic idea. That's it in a nutshell. Okay, so how can you use TFs to improve axon growth? Well, step one is you select a target. You choose a TF that you think organizes embryonic axon growth, but which is missing from the adult. Then you injure the animal, and then you inject to the brain a gene therapy vector that basically forces the adult neurons to produce the TF when they normally wouldn't. Then you trace the axons down the cord, and with some factors, what you see is that, yes, the presence of that TF in the adult neurons causes some axons to extend below the site of injury. And over the years, we've identified several TFs that have this property. Now, we know that TFs usually work in groups. So more recently, a major push in the lab has been to find combinations of factors to get even more growth. Dr. Venkatesh led this campaign to find TFs that synergize with one another. And earlier this year, we published a paper showing a combination of two factors that drove the most growth that we've seen so far. So we're really excited about this, and we plan to keep pushing on this approach. Well, this raises a couple questions. The first is, can a gene therapy approach really be translated into the clinic for spinal cord injury? And the point of this slide is that from a regulatory point of view, I think the answer is yes. The FDA has already approved gene therapy approaches for a variety of other sort of brain afflictions. So from that perspective, I would say, why not spinal cord injury? But there is a delivery problem that needs to be solved. What we've been doing is injecting these gene therapy vectors directly into the brain, and that's fine for proof of principle, but it's never going to work clinically for two reasons. The first is there are just too many neurons in too many places to reach with this approach. It's simply not practical. And the other problem is specificity. You can't control the spread of these gene therapy vectors. So you want growth from the injured neurons, but you don't want growth from the adjacent neurons that are involved in intelligence and emotion and so on. So how do you get your gene therapy delivery to be both widespread and specific? The solution that we're really excited about is the so-called retrograde approach, which we didn't invent, but we're certainly using. The idea is that gene therapy vectors can be taken up by axons and then transported backwards back to the cell bodies of origin. So the idea is, because axons come together in a very small volume in the spinal cord, with just one injection, you can get your gene therapy vector back to the widely distributed cell bodies that gave rise to those axons. And second, because only those neurons that project axons to that location in the spinal cord have access to the vector, you've got specificity as well. And this works. With Dr. Sulfas in Miami, we've been injecting retrograde vectors to the spinal cords of mice and then seeing which neurons take up the vector. We have a way to optically clear the mouse brain and then basically map and count the neurons. And what we see is that this gene therapy vector is reaching many different types of neurons all through the brain. And importantly, this is also true even if we inject the vector into the chronically injured spinal cord. So we see this as a very practical way to get gene therapy treatments in one shot and very specifically to the many different types of neurons that are responsible for all those different functions that I talked about that get disrupted after injury. And finally, how would gene therapy fit into the bigger picture? Well, if you start with someone who's suffered a spinal cord injury, there's often spared axons that span the injury. And we now know that by using stimulation or rehabilitation, you can get function from those spared axons. And that's a way to deliver immediate gains, today. I think along with that, by neutralizing some of these stop signals that I talked about, you can get additional short distance growth from some of these axons. You can make some relays. And combined with the stimulation, I think there's an opportunity, again, for short-term gains. So both of those exist. And I think the idea would be to push on both of those right now, today. And I think gains will be made from doing that. I also think the gains will be limited. And before too long, we'll come to a point where we're looking for more dramatic benefits from treatment. So additionally, I do think that some kind of a bridge is going to need to be built across the site of injury. And finally, where the gene therapy comes in, is we need to stimulate the ability of neurons to regrow their own axons. I think we could, for instance, deliver transcription factors to these injured neurons in this retrograde fashion, inject them right here above the site of injury. And by doing all those things together, the hope would be that we could induce more robust growth across the injury and affect some real functional gains. That's it. Time is short. So that was my final point. I really look forward to further discussion, and I very much appreciate the opportunity to present today. Thank you.
Welcome, everybody. Thank you very much to our presenters who are here before us. Michael Lane from Drexel, Murray Blackmore from Marquette, and Koffler from University of California, San Diego. Welcome, gentlemen. And Matthew, welcome as well. Very interesting talks. Everybody has their own approaches here, very distinct. We have cell transplantation approaches. We have boosting endogenous plasticity of existing systems, and then providing conduits, bioengineered conduits. Gentlemen, I guess I'd start this off by congratulating you on this extraordinary different kinds of work. But I guess I'd like to also ask a little bit of discussion about, number one, putting in these polymeric implants into animals with spinal cord injury. And then that is understandably like sort of what Murray is doing, inducing long-term regeneration of long tracts. And then Michael is, on the other hand, looking to transplant interneurons. Is there any way we can like just start the discussion about the distinct models and what their limitations? For instance, Kobe, let's start off with you. With the polymeric implants, when they don't dissolve out of it, they're still there. What's the thoughts about moving downfield about whether you're going to make the tube smaller or more biocompatible or even dissolvable?
Yeah, so the polymers are degradable. They're degradable over a period of time that we engineered. So the current mix is degradable over 9 months. So for example, when we looked after 6 months, about 50% of the material already degraded. It's important because when we started and we worked with different compositions and different materials, we thought about quick degradation. And when we had quick degradation over 2 to 3 weeks, yes, the material was not there, but the tissue also collapsed. So you need to have support over long enough time in order for the tissue to bridge for something that is strong enough and sustainable, that once your material is slowly degrading, the tissue can still hold by itself. So there is this balance that you need to put in place in order to have this, I would call, relay of modeling the tissue between the material and then the intact tissue that's then being moved.
And so, Murray, your approaches are suggesting that if we manipulate certain genes to either or promote regeneration, how does that speak to the audience, people who are looking at this in a sense that, can we get our own custom designed inhibitors or that we could use to use for certain genetic traits of people, or how does this work?
No, I'm glad you asked the question. It's the exact opposite. So my worldview is that there is an ancient and conserved through evolution sort of circuit of axon growth to the point that you can look at things like fish, even frogs, and identify genes that you absolutely need in order to grow a long axon. You can find the same genes in mammals playing a similar role. So I don't think we have to go down this route of sort of customizing the gene treatment to each person. I think there is a singular set of targets that would work well in a mouse, work well in a rat, and work well in every human being, certainly. Yeah, which I see as good news. I mean, thank God, right? Like, what if we did have to sort of I don't know, sequence your whole genome and then come up with the right genes? Like, no, it's not that. It's one shot.
And so with Michael, with your approaches, and you're able to put in these particular progenitor cells, how does that speak to genetic variability? And can you, is that something that is not a concern at this point because we're still trying to get graft integration? Or how do you see that playing down the road?
Yeah, when it comes into phenotype, it's a little hard to know. We don't know as much about these phenotypes across mammalian species, especially in the humans. So, whether these same interneurons serve the same functions in the human spinal cord. Right now, we speculate they do. We don't know for a fact. We're still learning. But we're kind of at the forefront of learning that these types of interneurons exist. And so we're really trying to establish what sorts of neurons there are, what potential there is for serving beneficial functions. And when we start to tease out subpopulations of these, hopefully we can get a cocktail of these different types of cells that'll all serve reparative functions. That's the goal. But we're still a lot to learn.
And so, Kobe, in your work with in San Diego, have you all, what kind of stem cells, so to speak? Michael has these specific progenitors. So what kind of stem cells are you putting in there? Are you thinking maybe you're going to be put other things in there to more specifically target lost pathways below the injury? Just curious about what you're all thinking about without divulging anything.
Yeah, so there's no need to divulge, So the data that I've presented in this presentation was using rat stems, and the idea there was to avoid immunosuppression and all these things that really complicate the survival of the cells and then make it more difficult to look at regeneration. The idea was to show the proof of concept that we can 3D print the scaffolds and load them with stem cells and implant them and look at biocompatibility. We now moved forward to more advanced preclinical studies. We're working on scaling up the technology, and there we're working with human stem cells from Mark Tuzinski. And those are neuronal stem cells, progenitor cells that we are further differentiating to more spinal cord phenotypic kind of cells.
Mm-hmm. Okay, so there is a movement to do that. And then I guess the other question, just as a follow-up, so these microchannels, as I said, I worked with them many years ago. Do you envision that? To me, when I see them, I can see that they're visible, so that to me means that they're big in the spinal cord. Are you thinking about how to make them smaller so that they take up less space, if you will?
Right, so that's a great question. What's the size? What's the appropriate size? Because an axon is 1 to 5 micron, let's say, in diameter. Do you want to make an effort to align each and every axon? You've got about 1 million axons in the spinal cord. Would you like to do that, or would you like to maybe align subsystems, because the axons, right, they're organized in subsystems? The basic idea that you have to remember is whenever you introduce an open channel, you're also introducing walls, right? So for every open channel, there's a wall around it. So there is a balance. If you have too many channels, you have too many walls. So what we're interested is what we're more interested in is how much cross-section is open for regeneration. So we're trying to optimize the wall thickness to the open channel, right? And that's part of the engineering part that we're working on, to make thinner walls, more channels that we can introduce. We're introducing 200 to 300 micron open channels. If you go higher than that, you go to 500, that's too much freedom for axons. And we'll go back, turn around. We're losing the whole aspect of linearity. So we need to stay in that range of the 200 to 300. We figure we don't need to go down too much because of the wall aspect, and we're just interested in trying to improve open cross-section that's available for regeneration.
And so let me just shift gears here for a second and ask you, Murray, with Michael's work and your work, is there any way that you could envision, for instance, even this is a different pathway that Michael's looking at, that the type of work you're doing specifically look at how to promote, for instance, these pathways that are important for phrenic breathing and stuff? I'm putting it out there just because I'm trying to merge two different technologies and see if it's possible. So for instance, I guess you can get stuff to grow beyond a border. Michael's trying to put stuff to grow into his graft. Is there some kind of linearity between that, without, not just on a paper, but principle-wise, that you could maybe coax things in different models the way you're doing it in the corticospinal tracts?
Absolutely. So I'll let Michael jump in on that after I'm done, for sure. But I mean, right off the top of my head, it's a variant of where I ended my talk. Now that we know that our retrograde gene therapy reaches cell types throughout the brain, that would presumably include the prephrenic neurons, the brainstem neurons that innervate the phrenic motor neurons. That was a mouthful.
Yeah.
My point is, why not try an injection of a retrograde vector right above one of Michael's grafts to see if we could coax more host growth into the graft to potentially innervate it. I mean, that would be the sort of thing that right off the top of my head would be like, yeah, you could do something like that.
Okay. And Michael, what's been your biggest, I guess, difficulty in getting these, is it just to survive even? I mean, I guess to get the host to come into it, it has to survive. Has it been difficult to get them with one population of mixed unique cell types?
So survival, we haven't really seen to be an issue with our donor cells. Firstly, the cells survive really well.
to Murray's point, not all neurons grow well in the adult mammalian spinal cord, which is why his gene therapy is so important. A lot of these axons don't grow. And so I think, to his points, I think gene therapy for respiratory neurons in the brainstem could be beneficial because it could coax those axons into the transplant. But I also think of it a little differently. I think that sometimes when these axons, like the respiratory axons that don't grow well, that maybe they don't need to grow into the transplant because we have spine-linked neurons. And the spine-linked neurons, as I said, are very plastic. So as it turns out, some neurons do have a pretty good growth capacity. And I think these spine-linked neurons, they do grow into the transplant. The spine-linked neurons we're transplanting, they also grow out. And so we can enhance that with stimulation. We can enhance it with activity-based therapies and rehabilitation. So we can improve the way they grow. We can't really coax more of those respiratory axons from the brainstem in very well. And so a gene therapy would be great for that if we need it. We don't know if we need it right now, but yeah. So the spinal neurons themselves have a great growth capacity. Gene therapy might work really well if we need those axons from the brainstem to grow to our transplant.
Okay. Kobe, I have another question regarding the polymeric applications. It's my understanding that your model is a complete transection model, which makes sense. And there were some diagrams how you can modify the pieces. Are you all considering, and I don't know how this could be, but an injectable solution, something that doesn't have to be so invasive and that maybe could be clinically relevant to be able to inject it into the spinal cord or into the cerebral spinal fluid? Is that something that you all are entertaining?
Yes, I was thinking about that about 10 years ago, and the technology hasn't been invented yet, right? Because the idea, what's important in my work is to have these linear microchannels. So it's not about the injectable, it's about injecting something that would take form once injected. That's the issue, right? And so not only take form, also be placed in the right axis, right? So it can take form, for example, and create channels, but if it goes perpendicular to the rostrocaudal axis, you lost all your effect. So it's definitely an idea that I've thought about before, but I still don't know how to approach that. There's a very sophisticated material engineering that needs to be there that I still haven't figured out. And if anybody has any idea, I'm happy to collaborate on that.
Right. Okay. All right. Well, thanks for letting us know about that, because there's hydrogels that are advancing now and all these things, but to keep a tube or a channel, that's what's the challenge in us, without the body trying to degrade it. So I guess I want to try to again unpack this for the audience. Where do you see, all of you collectively, where do you see, and I'm not giving the timeline, I don't want timelines anymore in my life, but where do you see the emergence of the very different therapies that you all are using with regard to, let's first and foremost, the field before you give it to the patients? Where is the stumbling block for each one of you in this translational spectrum, if you will? And again, there's no preference to talking, there's an open dialogue. Like, for instance, Kobe, I'm sure you have to get this stuff FDA approved, right? I mean, that's going to be.
Yeah, well, that would be a big hurdle by itself, because there aren't really any 3D printed devices approved for any indication. There's only one that I know of. There's a 3D printed hip by Johnson & Johnson. That's the first thing that came out, I think, last year. So clearing a device through FDA that would be a 3D printed device by itself is an issue, because FDA doesn't have an experience of that. But definitely the scaling up, so moving up from rodent models to more big models, large animal models, where we need the scaffold are just much bigger, right? So how do you scale up and keep your linearity and then load it properly? Loading scaffolds that are much bigger is not as I would say, easy as it is to load a rodent scaffold that is 2 millimeter long, 1.8 millimeter in diameter. So the challenges are those, engineering related challenges
Sure.
on the scaling up.
And then Michael, with regard to yours, I mean, obviously we have institutional protocols we have to adhere by, but let's face it, we would like eventually this work to go into humans. Inducible pluripotent stem cells, how far is that kind of stuff from clinical application?
Well, a lot of these cell therapies are already being translated. So neural stem cells and neural progenitor cells have been transplanted into people with spinal cord injury already. There's been several trials that have looked at that. And in Japan, there's trials that are now transplanting human iPS cells. So these are being translated. The one thing that's lacking in knowledge for those trials is they don't know which phenotypes of cells are being transplanted. So conceptually, the work we are doing can start to be translated into those existing trials relatively easily. Now, can we start to screen the sorts of cells we're putting in, and can we refine them a little bit to make them more effective so we don't get as much variability between patients like we get in our animal models? If we can reduce the variability, refine the cells we're putting in, and make them connect better, which is kind of conceptually what all three of us are trying to do, right? We're trying to get the networks to repair themselves in the appropriate way to the appropriate targets. We'll make the existing things that are already being translated, we'll make those better. Gene therapy is being translated now. So there are lots of things that are being translated for gene therapy already. I think something like Murray's treatment can be translated. There's going to be hurdles no matter what. And I think we try and figure out what they are as we move forward, but we need to try and move forward.
Let me jump in real quick.
I could ask Matthew to go ahead and field some questions.
Yeah, I want to roll up a few questions here, particularly probably first to Kobe, but to any of you. Anita says, actually back to the scaffolding question and the injectables, says, great point, Sasha. How would the scaffold work in people who don't have a transected cord? And then also someone earlier asked the question regarding Kent, actually, have anyone looked at hydrogel strategies similar to Matricel, or I'm assuming in vivo as well? And then lastly, Rob made a comment, there's now FDA guidance on 3D printed scaffolds. And even, one of the original presenters we had invited was Andrew Pelling from Spider Ward, who got a breakthrough device designation for his scaffold strategy. So rolling all those up together, Kobe first, maybe, how would you answer those comments and questions?
Yeah, so what I presented was transection in a rodent, because that would say the best way scientifically to test a question of regeneration. When you create a transection, you transect every axon. If something regenerates, you'd see that. You don't have questions of sprouting axons from spare tissue and so on. So that was the purpose of that experiment. Yes, we are now working. Part of the scaling up is to work with the clinically relevant models of contusions, where we 3D print scaffolds that really match every injury, right? Every injury looks different. Every injury is longer, or maybe have different contour, and we are printing those scaffolds to match specific injuries. So yes, we're doing that. The materials that we're using are hydrogels. So we are definitely doing that with hydrogels. With previous materials we used before were a bit more stiff, and so the interface wasn't as acceptable. I showed that reactive cellular, that it was just larger. That reactive cellular would prevent axons to regenerate through into the scaffold, into the implant. And part of the things that we achieved with moving into hydrogels was really the reduction of the reactive cellular that allowed host and stem cells that are implanted into the scaffold to really enter and exit the scaffold. So yes, I mean, that's part of what we're doing now with the scaling up to go into the clinically relevant models. And then that's the beauty of 3D printing, that you can match every injury.
Mm-hmm.
So I'll tag on to that, which I think originally asked by Jason, but I think to all of you, what about chronic injury models? How do you see? I know, Michael, you mentioned that, I think, towards the end of your presentation. Where does this intersect?
Yeah, we are actively trying to do that now. So the early studies and most of the work I talked about were in more sort of subacute stages, which in animal models are a week after injury, and we're delaying that further. So we're trying to look at transplants months after injury, and down the road we'll go further, just to see if we can see consistent effects. People have done transplants with neuroprogenitor cells across a huge spectrum, both people that I have learned from, as well as the Taszynski group has done a huge range of different transplants at different times. So they work at different times for sure.
Yeah, absolutely. I want to give a shout out to Paralysis, which gave us some seed money to form a collaboration, and we just went all the way. So we found a collaborator, a great guy, Stephen Perlmutter in Washington, to take our best gene treatment into a chronic injury model, so 3 months about post-injury, before they even get the treatment, and then to combine that with electrical stimulation as well, so to just kind of do the whole thing. We got some seed funding, like I just said, and then Craig H. Nielsen Foundation picked it up. So I just want to echo what Matthew said either yesterday or the day before. Chronic injury studies are harder in the lab, but man, if you think you've got something, the translation, the clinical testing is so much easier. All the problems melt away. Many of the problems melt away. So I think it's really important to invest in the chronic injury studies on the front end. It saves you time on the back end, is what I think.
And so Mary, being one of the, from my opinion, because I've known you so well through your career as a neurogeneticist, what would you like to offer, not necessarily with the work that you're doing, but just in generally, maybe even in the back of your head, like genetics, how do we see that moving forward? Is it that we'll transplant different types of cells, or that we'll just know what types of cells to put in which kinds of people? I'm getting a little bit bigger with the ethos as far as genetics. Is it more the interaction of what we're putting in together, or is it the nature of the recipient, if you will?
Yeah, I don't know. We live in the age of subdivision. We live in the age of, every person is different. Personalized medicine, cultural and scientific level. And I'm just going to push back. I'm just going to say, look, evolution figured out how to grow an axon hundreds of millions of years ago. Hasn't changed that much. It doesn't differ that much. And so I think there's an optimal cell type to put in the transplant. I think there's an optimal set of genes. There's probably an optimal stimulation paradigm. And maybe there's exceptions to every rule, but I think that's going to get you most of the way there for most people. That's my position. I hope I'm right, because the alternative is, again, so bad, I almost don't even think about it.
Right. Yeah.
Let me throw in another question here from Lana. How would any of these therapies work in people whose injuries resulted in actual motor neuron loss? Arguably the largest of all these therapies, since you're trying to increase either the descending input into motor neurons or increase potential of motor neurons. How important is it possible even, or is it possible even? Do you all three see the problem of a motor neuron restoration, generation, replacement? And just a quick heads up, we got a couple of minutes.
That's a really tough question. And so I guess it speaks to the fact that there's motor neuron loss at the level of injury, right? So it depends where your target is that you're trying to get axons back to. So if you're trying to get axons, you say you've had a cervical or thoracic injury, you need axons back to the lumbar level to improve locomotor function, sexual function, bladder function. That can be achieved because you haven't lost those motor neurons. They've lost their input. At the cervical level, though, if you're trying to restore cervical networks, that's much harder. And to Lana's point, you're losing motor neurons at the level of these networks that you're trying to repair. And so that is a pretty big limitation. We're putting in interneurons, they don't do the functions of motor neurons. Ideally, if you wanted to transplant motor neurons, it's conceivable. Some people have looked at that, but getting those motor neurons to grow to the appropriate targets is also very, very difficult. So it's a big hurdle when trying to repair networks that are at the level of injury. Good point, difficult problem to address, I would say.
Yeah, it speaks to
but I think that conceptually that's exactly where we all, working towards, right? We are working towards replacing the tissue with something that would be either as close as possible to what was lost, or to create some kind of a relay that would recapitulate whatever connectivity that was lost. So
optimizing function where we can, yeah. So improving the plasticity that exists with the spared networks that are there, and further enhancing repair to facilitate greater recovery.
Hey, one last general question, which has come up in a few different ways from Tracy, from Rob. What higher level? What are the roadblocks? What's slowing you down, or what needs to happen to pick up the pace of this particular research, or these strategies rolled up into one?
I'll throw out the old one. Because there are so many challenges that need to be tackled at the same time, it's very, very hard for any one lab to have the expertise to tackle them all. And I'm saying something specific. I think that when I put in pro-growth genes, I often can't see the effect of those genes because there's not a nice transplant to grow on. And we're funded to do transplants, but it's been really, really hard to make that work in my lab. Whole labs devote themselves to getting good transplants into injured spinal cords with variable success. So it's just really hard to have all the expertise in one place to do the full set of experiments that you need.
Michael, Kobe, quick.
I think that, it sort of relates to what Morris says. It's not only having the expertise, it's the fact that there's also really a lot of knowledge that we're still in the process of gaining about the system, right? The spinal cord is an extremely complex system just from the biology aspect of that. And we're still learning. So even though, we are presenting these wonderful achievements and we're moving forward to, preclinical studies and even clinical studies, there are still open questions out there that affect anything that we do. So I think it's important for people to know that we're also have questions ourselves, scientific questions that would then help us move things forward.
Michael?
Yeah, I think logistically, I agree. I think we need, I need people, because I have a lot of questions, like identifying these interneurons and figuring out which ones we need to put in. That's just one question. What types of stimulation or rehab we can use? Each question requires several people. So I need a lot of people to work together to answer those questions and move the answers forward, which will give us more treatments and optimizing those treatments consistently. But I think these things are moving forward. They're being translated stepwise, and we'll continue to improve on those things that are being translated.
Great. Thanks.
I'd really like to thank you guys for opening up like this and taking such questions. I know they're not easy to field. Matthew, go ahead. I'm sorry I interrupted you.
That's all right. Thanks. And thank you, Sasha, for moderating this. For each of you, just a side, there's a lot of questions that didn't get asked. So I would welcome you to check the chat. You can reply to folks that ask questions that, maybe were directed to you or you have an answer for. I think they'd appreciate it. And of course, we want to see you back at the breakout room this afternoon if you're able to make it. Love to have you there for a conversation.
I'll be there.
All right, so we're going to move on.
Gentlemen.
Thank you, everybody.
Yep, appreciate it. So now we're going to shift a little bit of gears, but kind of in the same vein. Our U2FP Sam Maddox is going to have a conversation with Bob Yant. Some of you may know Bob is a gentleman with a spinal cord injury, been around a long time, and he has been funding regenerative research for quite some time, and even so much so responsible for a company that you heard mentioned on the first day, Axonis. And so we want to kind of shift gears and from an advocate from the SCI community, get some context and perspective on what Bob's journey has been in funding this kind of research in an effort to translate it to appropriate clinical practice. So I hope we're going to be able to welcome Bob and Sam.
Hey, everyone. I'm Sam Maddox. I'm the director for the Scientific Advisory Board for U2FP. I'm really glad to be here with Bob, my old friend from the spinal cord injury path. And he and I both live in Southern California. Bob and I have had many conversations about scientific research, and I can tell you that 15 minutes isn't going to get it done for us today. We've got a lot of things we won't be able to say. But Bob, first of all, welcome. And you've been injured since 1981, back when Ronald Reagan was president, for goodness sakes. And tell us a little bit about your path, how you got involved in the whole cure movement, so to speak. And then we'll sort of segue into what you're doing as an entrepreneur and as an advocate.
Okay, thank you, Sam, for the introduction. I have a C5 spinal cord injury that from a beach diving accident in 1981. I immediately was I'd already had an interest in science, so it was a natural for me to take that interest in science to the science going on in spinal cord injury research. So I was a member of the board of directors of the group that was the American Paralysis Association, which started in the 80s. I was on that board for that group. Eventually became the Christopher and Dana Reeve Paralysis Foundation. I was on that board for about 30 years. As a member of the board of the directors, I received the scientific proposals that came in advance of the Science Advisory Council meetings for the individual investigator awards, and I attended dozens of those meetings. And I would read the proposals myself before going to the meeting, and I was always totally surprised at how wrong I was about what the proposals were that were funded. I also attended neuroscience meetings throughout the United States, and over the years I've met about 300 different researchers involved in some aspect of spinal cord injury research. And since my accident 40 years ago, I've raised approximately $20 million for spinal cord injury research. I founded a successful medical supply company, Cure Medical, which donated 10% of its net income to researchers working to find a cure for spinal cord injury. We sold that company in March, so now I'm able to donate to spinal cord injury research myself.
So Bob, back in the 80s, the story was that regeneration was finally recognized as a possibility. And then as we sort of moved towards that science area, as Dr. Blackmore explained, the emphasis was more on the extrinsic factors, these stop functions that stopped axon growth. Then along came some further work to show that maybe there's a way to get the intrinsic factor, basically to take as the example was, put the motor back in the cell. Your emphasis has always been on axons and axon growth. Why is that? What has led you to that sort of area of concern?
Okay, so I call spinal cord injury a condition of axonal disruption. And axons are the long extensions of nerve cell bodies. So my crude analogy is a tennis ball would be the nerve cell body with the string coming off of it, or the axon. So when we have a spinal cord injury, the axons are damaged. So the signals that go from the brain to the lower body about movement can't be transmitted. And likewise, these signals that come from the bottom of the spinal cord that take sensation up to our brain are also disrupted. So for the people listening to this, the people who have spinal cord injury are listening to this talk right now, if we want to get out of our wheelchairs, we have to regenerate axons. So earlier in my career as a volunteer fundraiser, I used to get excited about published papers showing recovery in spinal cord injury. But then some neuroscientists pointed out to me that the recovery was in sensory axons, not in movement or motor axons. And then I learned about the cortical spinal tract, which is this bundle of nerves that control hand movement and other movement functions. And that tract of nerves was discovered about 100 years ago, but it's also the most difficult tract to regenerate out of all the nerve tracts.
So take us a little further into the story. You were focused on axon regeneration, but tell us about this paper that came along that kind of really got you focused on the whole axon rebooting the function of an axon story.
So the word breakthrough is so misused that sometimes it can be meaningless. But this researcher at Harvard named Zhigong He made a breakthrough that caused, it's a true breakthrough in the sense of the word, it caused these cortical spinal axons to grow into, first of all, grow into the injury site and then grow through a little bit. And Dr. He, he's arguably one of the best neuromolecular scientists in the country or the world. And for example, on Thursday, he got nominated to be in the National Academy of Science. He's got 40 people in his lab, which is a large number. Graduate students and postdocs come to work with him from all over the world. And many of his laboratory workers come from the University of Beijing, which has been ranked by certain metrics as the #1 university in the world. So what he found was this molecule, P10, that naturally prevents nerves in the brain and spinal cord from continually growing. So P10 controls like the most basic parts of growth in the central nervous system. And there's never been a more powerful or important gene than P10. And now researchers are working to realize the potential of inhibiting P10.
So you took this a little bit further, though. You saw this paper that he first published about P10 and how it was able to restore some pretty dramatic axonal growth. What did you do?
So I always wanted to start a biotech company to bring a treatment for spinal cord injury to market. But there was never a potential therapy that existed that really was worthy of starting a company. Sorry, I'm tilting over here. I get dizzy in the morning. But when the P10 breakthrough was made, I saw the possibility for a treatment that could finally be effective in humans. So I was concerned that if I didn't patent that P10 molecule, that a big pharmaceutical company would come along, secure the research, and if that didn't work out, they would just put it up on the shelf. And I also didn't want to have potential investors who were just more concerned about making money and not bringing this treatment for spinal cord injury to the people that needed it.
So this company Axonis, last year, your CEO, Joanna Stanica, presented information about another therapy that's maybe going to come before P10. P10s continue to be working. And we don't have a whole lot of time left, but I do want to ask a couple questions as from the point of view of being an advocate. What can you tell other people who are in the spinal cord injury world who want to learn about the science, want to keep up with what's going on without having to go to get a graduate degree in neuroscience? How do people do it?
Okay, there's several ways. First of all, don't believe every scientific, every press release that comes out about a cure for spinal cord injury. You got to stay in the learning mode. So that means learn the anatomy of the spinal cord, learn the anatomy of neuroscience. So when you see a word, if you don't understand, look it up. And then there's a great article on the internet called How to Read a Scientific Paper. Search that for Google and learn how to read a scientific paper. I would say read Sam Maddox's posts and explanations as you're doing right now. Attend U2FP annual symposiums. Look at which researchers are funded by the NIH and the Craig H. Nielsen Foundation, because those projects have undergone peer review, which is a really, really important thing that scientific papers get judged by the fellow reviewers. Get to know neuroscientists. When you have a question about a particular paper or an area of research, it really helps to get the perspective of a neuroscientist who works in the field. And finally, attend other scientific meetings besides the UFP meeting. So sitting through all these talks can be like strenuous and at times boring, but scientific meetings are a great place to meet scientists and get to talk to them off the record.
One more thing, Bob. how do you keep going after 40 years? A lot of people get excited about cure and after the first 5-year promise is expired, they drop out. How do you keep going?
As much as all of us want to get out of our wheelchairs tomorrow, to stay engaged, you got to look at this in the long-term perspective. In my 40 years of injury, I've seen tremendous progress in the field of spinal cord injury research. We're now at the point where there's a dozen more companies trying to bring various treatments for spinal cord injury to market. These companies didn't even exist a few years ago. Clinical trials can take 7 years. So we have a long time to go before a potential drug reaches the market. The most important thing is to never give up. Listen, as a fundraiser and a research interested person, I always used to get the question about when will a cure for spinal cord injury be developed? I learned early on not to ever answer that question because I don't know the answer. But if we don't continue to support science, spinal cord injury research, I know the answer of when there will be a cure for spinal cord injury. The answer is never.
Great, thank you.
Thanks, Bob. Thanks, Sam. Have great respect for your longstanding advocacy work. Really appreciate it and appreciate you joining us today. I want to add a couple things to what Bob just said, and one of them is, just in the last few years, we've added almost $20 million to that research effort through our legislative passages of bills. And a byproduct of that, and I mean, I shouldn't even say it's a byproduct because in some ways we feel more important than the money, is exactly what Bob said, is advocates, people with spinal cord injuries who become savvy, who become knowledgeable about the science and are growing in their relationships with scientists as a result of these efforts in Minnesota and Washington and Pennsylvania and Ohio. And we hope to pass 3 more in the next year or two. So again, thanks so much, Bob, for being here. Appreciate it. Thanks, Sam, for the conversation.
Kudos to U2FP for all that you guys have done. Keep it up.
Thank you. Okay, now, so we're going to switch gears a little bit. And the remainder of the day will be focused on spinal stimulation, pivoting a bit from our regenerative strategies that we've been talking about this morning, but very important. And obviously, many of you are aware of some of the developments of spinal stimulation. We're going to hear from some leaders in that field. And again, I want you to be kind of having your context head on and your strategy head on, because you're going to hear a variety of points of view, a variety of places where spinal stimulation sits. And always in the back of our minds is thinking about, okay, what can that be in combination with? And so even as you heard in some of the presentations this morning, Corna nods to that from Murray, nods to that from Michael. How can some of these regenerative strategies be combined with spinal stimulation? In fact, Murray even talking about the project with the collaborator using that retrograde gene therapy alongside spinal stimulation. So now we're going to pivot. Please welcome Uzma Samadani, and you'll hear one of my little intros. Now, please welcome Dr. Uzma Samadani, physician and researcher in TBI and SCI at the University of Minnesota and also the Minneapolis VA and in private practice. She will be talking about the provocative question as to whether or not we are ready to deploy epidural stimulation in the clinic, albeit continuing to research and investigate both mechanism and clinical effect. So please welcome Dr. Uzma Samadani.
I'd like to thank Unite to Fight Paralysis for inviting me to give this talk. This is a huge pleasure. It's something that I feel very, very passionately about because we have been able to achieve some great outcomes for our patients, and I hope that we can continue to work on this in the future. My topic is epidural stimulation for spinal cord injury. Are we ready for non-research deployment? I'm in private practice. I also work at the VA, and I'm affiliated with the University of Minnesota for research purposes. These are my disclosures. The ones that are really relevant to spinal cord injury are that I have had funding in the past from Abbott, but it's all been related to serum markers for brain injury. And I also have had funding from Medtronic related to spinal cord injury, but it's not related to this topic. This is what a spinal cord stimulator looks like. So what this is a lead that's hooked up to a battery pack, which is called a pulse generator. And they are placed underneath the skin surface. It's an operation that takes less than 2 hours. It basically takes about half an hour to do, but then we also have to do what's called monitoring to make sure that it's in the right spot and it's stimulating the nerve roots correctly. So the whole operation, the time in the operating room is about a little over 2 hours if you include anesthesia and monitoring. And so this sort of graphic is what the end result is like. And like I said, we can do this on an outpatient basis. It's not an inpatient surgery. It doesn't require an overnight stay. It does require being put to sleep, which is general anesthesia. So who all is doing this? Well, we're doing it at the University of Minnesota. The clinical research trial was started at Hennepin County Medical Center, and we've also done it at the VA. And then off-label, which is part of my private practice, I've done it in about 10 patients. There are other centers that are doing this as part of clinical trials at UCSD, at Mayo, University of Kentucky, and UCLA, among others. So our first paper that we published on this basically showed that in 2 patients who were both women, we were able to achieve volitional movement. And what that means is that the patient was able to move their legs by controlling them, by thinking about moving them. So this is, something new for a patient who has otherwise been, chronically paralyzed. Our second paper that we published was in the first 7 patients, and 4 of these were able to achieve not only movement, but movement that they were able to maintain even after the stimulator had been turned off. So that's, volitional movement in the absence of stimulation. And what that means is that the pathways were always there, and the stimulation allows the spinal cord to sort of remember how to transmit that signal. And then when we turn the stimulator off, the spinal cord still is able to do that. So, it suggests that the capacity for recovery might be greater than we previously thought. And other people have done studies on this to suggest that this makes sense. This is the data from that second paper. So the patients ranged in age from 30s to 50s. They were several years post-injury. So the mean time post-injury was 7.7 years. Most of them were completely paralyzed, meaning they had no motor, no movement, and no sensation. Most of them had their injury site in the thoracic spine, and their vertebral fractures at relatively same levels. Some of them were falls, 3 of them were car accidents, and 2 of them were sports injuries. And then we placed the stimulator via a surgery at those particular levels. So I performed, I think, the first 14 surgeries that were done for this operation. And so in these first 7 patients, you can see what the spinal cords looked like before we placed the stimulator. And what's pretty clear from this is that the spinal cord was pretty thinned out. And sometimes it actually thins out both above and below the injury. So we didn't know what the capacity for recovery would be with people who already had, a fairly thinned out spinal cord. What that means is that the spinal cord hadn't transmitted signals for such a long time that it had actually atrophied, and it had sort of lost the bulk of myelin pathways along those nerves. So this is an example of one of the patients. This is patient number 3, and she's on a stationary bike. And basically what you can see is that she's capable of driving the bicycle, of her own volition. So, this is pretty impressive for a patient who's been paralyzed for years. And you can tell by the muscle atrophy that, you know, she's probably had chronic weakness in her lower extremities for quite some time. So we're very optimistic that, you know, we've actually been able to achieve some level of biking in almost all of the patients. Now, let's go to the next slide. This is a summary of the data from the 3 best patients out of the first 7. And what you see in panel A is the patient's leg movements with the stimulator off. And what you see in panel B is the patient's ability to move their legs with the stimulator on. And so each of these colors represents a particular leg movement. So this sort of rusty orange represents plantar flexion of the foot, and the red represents dorsiflexion of the foot. So with the stimulator off, you can see that this first subject wasn't able to do very much, but with it on visits 9, 10, 11, and 12, they were able to do quite a bit. And similarly, you can see that, you know, for each of the muscle groups, you know, so for example, for plantar and for flexion and extension of the hip joint, you can see quite a difference in activity in some of the subjects, you know, with the stimulator off versus on. Subject 3 actually had the best improvement with the stimulator on. So it's actually quite dramatic, you know, when someone is able to achieve movement after being chronically paralyzed for quite some time. And that's what makes us so optimistic about this technology. So now let's talk about using it out of the research setting. Well, we've had 18 research devices placed. We've gotten volitional movement, which means the ability to move in almost all of the patients. At least 4 of them are able to move even after the stimulator's turned off. We've seen improvements in bowel, bladder, and sexual function in some of the patients. And we've seen improvements in spasm and back pain in some of the patients. So now let's talk about the ones that were done sort of not as part of the clinical trial. So of the approximately 10 devices that I've placed off-label, 2 of them have had poor outcomes. One had the worst outcome, where the device actually became infected, and the patient had had the surgery in Minnesota, but then had returned home to the West Coast and actually had to have the device removed there. And this was the only patient who was transiently worse after the procedure. So this was a gentleman who had come to me, and he actually had some level of improvement in his lower extremities, which made him not a great candidate for the clinical trial because he didn't meet inclusion criteria. But unfortunately, he actually got transiently worse. He subsequently got better. So that was our worst outcome that we've seen. And then we had another patient where the patient was extremely thin, and the pulse generator, which is the battery pack, looked like it was going to dehiss through the skin and cause a skin breakdown. And so I actually removed the device in that patient because I was concerned that the patient would develop a skin issue. And that patient didn't have any deterioration in exam or anything like that, but it didn't make sense to keep the device in if there was a very high risk of causing a permanent skin breakdown. The 3 challenges that we've had in the off-label placement of devices, I would say, are insurance coverage, infection and skin issues, as I've just mentioned, and then optimization of programming. So how do we overcome these challenges? Well, in all 10 of these patients, we've been able to have their insurance cover it. 6 of them were VA and TRICARE. And so the VA actually has some flexibility with regards to spinal cord injury. 4 of them were private insurance, and some of the insurances will cover stimulator placement for what's called complex regional pain syndrome. So what that is it's a description of symptoms, a constellation of symptoms that sort of are known to respond to spinal cord stimulation. And those symptoms consist of things like pain in the lower extremities, in this particular case, changes in skin, hair, or nails, unusual swelling or edema, weakness in the lower extremities, which all of our patients have had, contractures, which most of our patients have had, and atrophy, which all of our patients have had, and as well as pain. And the pain doesn't necessarily need to be in the lower extremities, it can be in the back. So if you have a constellation of those symptoms, we can submit to your insurance for coverage. And like I said, United and Blue Cross Blue Shield have both covered this in the past for at least 4 patients. So I'm optimistic that we would be able to get insurance coverage for other patients. It's a little bit trickier because you do have to, document that you've tried conservative measures for the treatment of complex regional pain syndrome. But if you can document that, then it's possible to achieve insurance coverage. Now, let me give you a case example. So unfortunately, most of my patients were done at the VA, and I can't talk about their cases because of the VA has fairly extensive HIPAA requirements. So I'm only going to talk about one patient who's given me permission to use his images. And the patient's name is Brad Baker. He is a former ESPN X Games racer. He's a national champion. He won the Pro Flat Track champion at 20 years of age. And this is what he looks like when he's on his bike racing. And he came to me on July 22nd, 2018. He flipped over his handlebars. And actually, I wasn't on call, but one of my colleagues who works in the emergency department actually tweeted me on DMs and said, hey, is there any chance you're around? And, could we talk? And he knew that I felt very, very strongly about early intervention for acute spinal cord injury. So he reached out and I was able to come in and cover the case. This is what Brad Baker's CT scan looked like after he had flipped over his handlebars. And what you can see is that this is T6, the thoracic 6 level, T7 and T8, is that he has fractures at T6, 7, and 8. And you can see that there's a big piece of bone right in his spinal canal. And then the T8 just is an end plate fracture. He also had, rib fractures and transverse process fractures, which are the bones off to the side. But you can see this burst fracture, coming through that vertebral body and crushing the spinal cord. And really that was where, the worst of his injury was. So I ended up taking him to the operating room that same day. And this is what we did. So we did this repair. And the patient, Mr. Baker, did actually surprisingly well. So he went from having almost no feeling in his legs to having about 15 to 20% feeling. And then he also had sensation mostly through his bottom, which he described as closer to around 50% sensation. And then he had trouble knowing where his legs were in space. But sometimes he was able to tell where they were, particularly if he looked at them. And then he did have some movement that he was able to get back in his hip flexors. And so you can see him participating in therapy. This is a couple of months after the injury, about 8 months after the injury. And you can see that he's starting to be able to move with a walker. His recovery then plateaued and he didn't really improve further. And I saw him about 18 months after the injury. And at that time, he didn't meet criteria for participation in a clinical trial because he had some sensation and movement, but he wasn't fully recovered. He wasn't able to stand on his own or walk on his own. And so we talked about it and we ended up placing an epidural spinal cord stimulator off label, and his was covered by insurance. And this is him now 3 years since the accident. And unfortunately, I don't have the video, so I can't make it play, but he's able to stand. He's still not able to walk without braces or a walker, but he's slowly and gradually improving. And, I talked to him recently. He's now 20 months post epidural spinal cord stimulation. He's back on his bike, which tells you what kind of personality he is. He's a phenomenally resilient person. This is him doing a wheelie on September 8th. And he's able to stand. He's still not able to walk. He's using leg braces. He's having trouble with the stimulator being inconsistent. It's position sensitive so that it only is giving full stimulation to his spinal cord if his back is completely straight. That's something that we now know how to fix. So, as I said, each case is very different and we're working on, working out these kinks. And the other problem that he's had has been with targeting specific muscle groups. So if he wants to engage his quads to lock out his knees so he can stand, he's finding that the stimulator also engages his calf muscles, and it puts him up on his toes. And then it also engages his hip flexors, so it makes it harder for his hips to move forward. So how are we overcoming that? Well, basically, the nice thing now is that Abbott in January of 2021 came out with this Neurosphere device so that we can remotely program. And our hope is that with remote programming, we'll be able to tailor the stimulation in a more subtle way, because it turns out there's quite a few ways to program these stimulators, and that makes it challenging. And so we're working with our colleagues at University of Minnesota, and we're working with physical therapists, throughout the country to try and figure out the way to optimize the stimulation. And I think that is by far the biggest challenge for this patient group, is how do you get the benefit out of the stimulator after you place it? And so that's really where we're focusing most of our effort and energy right now. So ultimately, is epidural stimulation for spinal cord injury a good idea for patients outside of a clinical trial? I would say you have to weigh the pros and cons. The pros are that it's a relatively low-risk surgery. It's done as an outpatient. It takes less than 2 hours, and the device can be turned off or it could be removed if necessary. The cons are 1 patient out of approximately 28 actually was worse after, although it was only transient and they did improve. Some have shown minimal or no benefit. So I can think of at least 2 patients where the benefit was pretty minimal. 1 basically only had improvement in spasticity, and 1 felt that he didn't really have any benefit at all. And then again, optimization remains the biggest challenge. So, I think that as we do more and more of these cases, patients are going to be doing better and better. And that will really enable us to tailor the stimulation and the optimization to help as many people as possible. So the team that works on eStand is huge. Ann Parr, Dr. Ann Parr, Dr. David Darrow, and then medical student now, Dylan Sturdivant, professor at the university, Tay Nedoff, Aaron Phillips and Andrei Krasiokov, they're in Canada. Patricia McCracken she's actually a colonel in the military who works in our lab at the VA. And Aliyah Ali, she came to our lab as an undergrad. She's now a postbac. That's the team. David Balzer, Dr. David Balzer is a rehab doc. He started in my lab as a medical student and stayed. Now he's almost done with his residency. I think he finishes in the following year. Caleb Hoover's now a medical student, and Shivani Venkatesh is now a medical student. So we've had an amazing team, and I give credit to them for all of the hard work that they've done. We've been funded by Minnesota State Office of Higher Education, and then we've also had devices donated by Abbott. And so, obviously we're grateful to them for that. If anyone's interested in learning more, they can please feel free to email me. And these are my phone numbers for both non-VA and VA patients. Thank you very much, and I'm happy to take any questions in the discussion.
Now, welcome Keith Tansey, a physician and researcher at the University of Mississippi Medical Center in Jackson, VA. And in response, you can maybe tell where this is going. Dr. Tansey is going to talk quite a bit about the mechanism behind spinal stimulation with a focus on transcutaneous stimulation, but also really some critical questions about how researchers profile patients, how they measure effect in patients, and what we really understand. And I hope this is also provocative. So please welcome Dr. Keith Tansey.
Hello, this is Keith Tansey. I'm presenting today on neuromodulation. As usual, things are more complicated than that. This is work that was carried out at Shepherd Center in Atlanta and then analyzed in the Center for Neuroscience and Neurological Recovery at Methodist Rehabilitation Center in Jackson, Mississippi. So after a spinal cord injury, there's a new anatomy and a new physiology. As you can see here, the inputs from supraspinal centers, which are impacted by the spinal cord injury, and sensory input from the periphery meet in the spinal cord and are integrated to generate motor or autonomic output. And after spinal cord injury, there's a disruption of some of this descending input and ascending feedback, but also the sensory input is no longer normal we found in our lab, and others have described as well, how the sensory input is changed. Yet this still represents an access point to the spinal neural circuitry to try to augment motor or autonomic output after spinal cord injury. So I'd like to think about neuromodulation in the sense of restorative neurology. This is intervention designed to target the uninjured or altered nervous system that remains after spinal cord injury. And we will speak today about electrical neuromodulation in an attempt to modify an output behavior. In this case, we're going to talk about gait. And the way we know that this can work is because stimulation can generate activity in large sensory afferents that come from the periphery, carrying information from skin and joints and so forth. And they project into the spinal cord to the neural networks that are there. Specifically, stimulation of these posterior roots or these large sensory afferents can activate directly motor neurons in the spinal cord to cause reflexes if stimulated with enough strength. And today, the work I will be speaking of uses transcutaneous spinal cord stimulation or spinal stimulation with a non-invasive approach. The disadvantages of this approach is there is not as much spatial resolution as there is with epidural electrodes, but the structures that are activated are the same in that the reflexes look exactly the same with epidural and transcutaneous stimulation. This is a non-invasive process, therefore it is much cheaper and without the surgical and anesthetic risks. And this allows study of stimulation intensity, pattern, frequency, and so forth much more readily in many more individuals given the same budget. So when the large afferents are stimulated strongly enough to cause a reflex, it turns out that the response depends on the frequency of stimulation. In the top panel, you can see individual reflexes being recorded in the quadriceps and hamstrings muscle when the stimulation happens twice a second. However, when that stimulation is increased to 30 times per second, you can see an interesting phenomenon, and that is that first shown here in the quadriceps, there's a period of time during which the reflexes cannot be expressed, and then an envelope where they are expressed, and then they're not expressed again, and then the envelope is again expressed. And this alternates with activity in the hamstrings muscles, its antagonist during stepping. And this pattern of flexor, extensor is occurring at the frequency of stepping. And we think this is an indication that the interneurons in the spinal cord are converting this tonic stimulus into phasic output. Now, there are several assumptions for how electrical neuromodulation might be accomplishing some of this work. There's a fair number of folks who are talking about the changing of central state of excitability to increase or suppress motor output. But as you'll see later in this talk, this is an overly simplistic idea. But the thinking is that sensory inputs that might not be sufficient to activate motor outputs can now be augmented by stimulation with an increase in this sensory excitability, and therefore motor outputs can be expressed. The other question has to do with whether sensory stimulation of a tonic nature can generate phasic output of spinal circuitry using this intraneuronal circuit to get locomotor output. And if you can see here, this is work done in the 1990s by Dimitrevich and colleagues. It showed is as you change muscle strength, stimulation strength, this is only one stimulation strength shown here, or stimulation frequency, you can fine tune a tonic input to generating tonic outputs all the way to generating a nice phasic output. And when I was a graduate student and a postdoctoral fellow, this was the proof that I needed to demonstrate that there is circuitry within the spinal cord that can convert tonic input into phasic output, and that there is a central pattern generator in humans just as there is in animals, or can be. What you can see here is alternating activity in the quadriceps and hamstrings, the upper leg muscles. And you can see phasic output in the lower muscles, the tibialis anterior and the soleus. But you should note that the activation of these two antagonists is synchronous. So the rhythmicity is present, but the pattern isn't quite right. And in reviewing all of the records of complete spinal cord injured subjects that Dr. Dimitrevich and colleagues looked at, the prevalence of being able to generate this kind of output with tonic input was actually not as common as originally hoped. More like 11% of those records showed this ability. So the spinal cord has the ability to generate this rhythmic output in the context of phasic output in the context of tonic input, but not easily. Now, the other ideas that have come from earlier work was that there were certain stimulation frequencies that could be used to augment things. This would be a case of fixing the weak, taking limited input or limited output that's in the face of sensory feedback and some descending command, and that doesn't generate any output, and converting that into output by adding tonic stimulation to this phasic sensory feedback and whatever descending command there might be left. And this was thought to occur best at 25 to 50 hertz. There are some folks, however, who may not walk well, not because they're weak, but because they're in fact stiff. And there is some excitement about decreasing spastic motor behaviors at higher stimulation frequencies. And the idea would be that without much activity being put into the system, there's tonic motor output. But when you add the neuromuscular stimulation, you may actually get rid of that tonic output and get rid of the spasticity or overactive motor function. So the study that we conducted was to look at the effect of stimulation frequencies on neuromodulation of motor output during robotic guided and measured stepping. In this study, we allowed the stimulation frequencies to randomly vary from 1 to 100 hertz every 6 to 10 steps during robotic guided stepping, and measure the changes in the robotic forces needed to maintain that stepping. Those forces were compared to the forces needed without stimulation. All the stimulation strengths were less than the strength to stimulate reflexes, rather designed to activate circuitry in the spinal cord. And all the participants had motor incomplete spinal cord injuries. And as I mentioned, this was graduate work of Jason White and the data collection, and postdoctoral fellow work of Matthias Krenn in the lab there in Jackson, Mississippi. So how did we actually do this work? Well, we used a Locomat, which has force actuators at the hips and knees. And not only can the robot move the legs through a predefined trajectory of physiological gait, it can report the forces that it's using to maintain those positions. So you can see that the desired angles of the limbs through time are matched well with the actual angles, and we can measure the forces there in the actuators. So here's an example of the forces that are recorded in the hip and in the knee during stance in black and swing in gray with no stimulation, 89 hertz stimulation, 25 hertz stimulation, and no stimulation. You can see these forces change relative to their baseline, and we can measure that change. And yet the position control is maintained throughout this stimulation. So we studied 9 individuals, all of whom had motor incomplete Asia impairment scales of C or D, spinal cord injuries, with a variety of neurological levels of injury at times as early as half a year out from spinal cord injury to 5 years out from spinal cord injury. And what we did was we had these subjects in the robot walking with 50 or 60% body weight support at 2 kilometers per hour for 310 steps. And during that time, we could record the joint angles, the joint forces, and the electrical activity in the muscles. So here's an example of the 300 steps being recorded and the randomly selected stimulation frequencies and the forces at the hip and at the knee during swing in black and stance in black and swing in gray. And what you can see plotted down here is how those forces in stance and swing changed depending on the stimulation frequency. In this example, as the frequencies increased, the robot produced more force in both hip and knee during stance, and likewise produced less force during swing in hip and knee. So shown here is all of the data of all of the subjects. And one of the first striking things you can see is that the robot either produced less forces during stimulation in swing, all these blue boxes, or more forces in swing, all these orange boxes. So in 4 of the subjects, the forces were decreased during swing, and in 5 of the subjects, forces were increased during swing. At the same time, there were, in the ones that had decreased forces during swing, there was a predominance of increased forces during stance. Likewise, in the ones that were increased during swing, actually had decreased forces most often during stance. So this stance swing difference is the first report of a differential effect, both across the patient population and in the 2 phases of gait. Nowhere did we have any individuals where all the boxes were blue or all the boxes were orange. We were able to group these patients using an algorithmic clustering program, and we found these 2 were very blue, these 2 were less blue, and practically no orange over here. These 2 had a little bit of orange over here, and these 3 had lots of orange and more blue than these other 2 up here. So we grouped those, and then we went and looked at what was the effective stimulation frequency in these different groups. And what you can see is in the groups A1 and A2, where there was more force provided in stance and less force provided in swing, that as the stimulation frequencies went up, the absolute change in forces also went up. On the other hand, in the other group where forces were less in swing, then there was this inverse relationship between stimulation frequency and the amount of force changes. It was sort of a sweet spot in the 20 to 30 range for getting the greatest motor output. But at the same time, there was another group, 2 out of the 9 patients, where the effective stimulation frequency was really none at all. So we have 3 out of 9 that do this pattern, 2 out of 9 that do this, and 4 out of 9 that have this spasticity suppression kind of a profile for stimulation frequencies. So here's an example of that suppression of muscle activity at different stimulation frequencies. This is with no stimulation. You can see activity, including clonus, in these muscles, tibialis anterior and soleus muscles, that as you increase the stimulation strength, increase the stimulation frequency, these muscle activities go away. The same thing can be seen up here in the hamstrings. And with no stimulation, this activity returns. In the B1 group, here's an example of gait muscle activity being augmented by stimulation frequencies that peak in the mid-range, but at lower or higher frequencies, there's less augmentation. So a small amount of activity increases here at the best frequency and then decreases again. Same thing with hamstrings here, and with the 2 lower leg muscles here being the greatest envelope of EMG activity during these mid-range frequencies. Finally, we can see that there can also be augmentation of pathological output in the same sort of pattern. Here's a patient with not much motor output, some clonus in the soleus, but when these stimulation frequencies were increased, the amount of clonus got greater and greater, and then lesser and lesser as the frequencies went up and then back down. So the conclusions that we have show that there is a spectrum of frequency-dependent motor profiles. Half of the patients approximately use the robot less during swing during stimulation, the other half use it more during swing during stimulation. And it really mattered as to the effects of stance versus swing, as opposed to an effect that happened across the entire gait cycle. By the same token, frequency cannot always change that spinal cord output. The group B2 didn't respond much to changing frequencies. We did have that one group in B1 where 3 patients, in fact, had this pattern of increased motor output at mid-range frequencies and less at higher frequencies. But with the high variability of output, it's important to know in whom and how we're applying these stimulation protocols. And we need a better way of physiologically profiling these patients. None of the parameters that we measured, time since injury, level of injury, severity of injury, really was any clue as to who was going to respond and not respond, and whether they were going to have the suppression of spasticity or augmentation of physiological stepping. So we need to find an algorithm for finding and adjusting optimal stimulation parameters better than randomly testing in each individual, or worse, just applying the same stimulation in everybody. So I recommend that we tell patients with neuromodulation. One size does not fit all. And there will be some who benefit and some who are potentially could be made worse with worse clonus when stimulated. We need to define this method for profiling patients so we know what type or class they're going to be before everybody gets implanted with a stimulation device. We need to consider conducting non-invasive trials with transcutaneous stimulation, just like we do with baclofen and spinal stimulators for pain control, rather than submit everybody to a surgical implantation before we know what kind of response they're going to have. And it's another argument to be making measurements of gait forces, not just looking at EMG or videos in terms of assessing the effect of the stimulation. Then we have to do a much better job of studying patients after a stimulator implantation to determine if the correct settings and the correct dosing is consistent day to day and persistent over time, and whether there's any side effects that accumulate or diminish over time, and then adapt those stimulation strategies. And we have to continue to work on improving our understanding and insight. Proof of principle does not equate to standard of care. We get to that more deliberately, but ultimately successfully. So I thank you for your attention.
Now welcome Mikailo, the CEO and co-founder of Anuvo, with a PhD in bioengineering at UCLA. And their company Anuvo has, in 2019, received FDA breakthrough status, breakthrough device designation status. And now we're going to have a conversation about bringing discoveries like this to the commercial marketplace as a product. Please welcome Mikailo.
Hello, everyone. My name is Mikailo. I'm the CEO and co-founder of Anuvo. Anuvo is a biological medical device company. It's my honor here today to talk about the journey that we've been through to commercialize our product for restoring functions after ACI. Let's get started. I know that both the company and myself are quite new to you, so let me do a brief introduction. I got my PhD trained in bioengineering at UCLA with a focus on neuroengineering, and I also have a background in microelectronic system and wireless communication. Professor Yant at UCLA, he's my co-founder, and he's also leading expert in the field of neuromodulation. We founded the company together in 2016 with a goal to improve the quality of human life through the technology that we developed. Before the formation of the company, we have accumulated decades of experience, knowledge, and insight in developing neurotechnologies and devices. Through the support of the federal government grant from like NIH and NSF, we invented different technologies to build a retina implant to help the patient restore, the blind patient restore their vision. We developed technologies for cortical implant, for high-density neural recording, and we even developed epidural spinal implant for ACI application. And most recently, we've been working on gastrointestinal implant to restore the gastrointestinal motilities. And those key technologies and know-how that we developed at UCLA, they become the foundation platform technology of the company. As of today, we have a small team of 9 people with complementary expertise. Our first product to treat paralysis due to spinal injury has received FDA breakthrough device designation, and we are currently conducting our FDA regulatory trial in the US. To date, we have raised around $8.5 million from both private investor and the non-dilutive SBIR funding. And we expect that more resources will be available soon to help us speed our product development to help the patients in need. We are interested in neuromodulation, trying to use modulation to treat chronic disease and injuries that are not curable or treat effectively using a conventional pharmaceutical approach. And we are particularly interested in spinal cord injury. As you know, patients with ACI often lose their sensation, mobility, even autonomy functions completely or partially at the level of the spinal cord region. But the neural circuitries both above and below the injury segment, they might still remain intact. And there's also evidence showing that spinal cord stimulation, they can be a potential treatment for ACI. The principle behind this is that the spinal cord stimulation, it can modulate the dorsal sensory roots, and then along with the descending brain command and the proprioception feedback from the limbs can all work together to drive the motor control, as shown in the simple animation. So We first started on the spinal injury project from our preclinical research. So our role is to develop epidural implant for ACI application, and this is an NIH-funded project. We take up this problem from our perspective and background as neuroengineers who can not only develop technologies, but also understand the underlying biological mechanisms. We are intrigued to know, for example, how many electrodes are needed to make this, spinal cord stimulation more effective for motor function recovery, and what waveform should use. Should each electrode deliver the stimulation at different or the same timing, and how flexible should the electrode be, and how small the size of the implant should be. And we not only ask questions, not just do brainstorm on paper. On the right-hand side is the outcome of our project. So at the end, we developed a very integrated and miniaturized epidural implant with only 0.5 centimeter cubic size, and the weight is less than a gram. This device is controlled by a wireless graphic user interface. So the tiny microelectronics in the middle here, I can support up to 160 channel neurostimulation, 12 channel recording, and the wireless power and data link. And we also developed a method that can connect the hundreds of stimulation output to a very high density epidural electrode array, and the array is made by thin film technology with only 20 micrometer thickness. We then expand our interest to translational research. We are interested in seeing how spinal cord stimulation, either non-invasive or invasive approach, can help the ACI patient to regain their voluntary motor functions. There's a hypothesis behind this. Imagine that there's a threshold for the spinal cord circuitry excitabilities. Intact, healthy person, they can easily exceed the threshold to initiate their motor functions, but the injured cannot reach the threshold at all. However, the hypothesis is that through spinal cord neuromodulation, it can modulate the spinal cord circuitry into a physiological state that can enable voluntary motor control after ACI. We then gradually started to pay more our attention on the non-invasive approach. Here's a research done by our collaborator, Dr. Sasha. He's the methodist. So tonic periodic non-invasive spinal cord stimulation is applied through by placing one or two non-invasive electrodes on the back of the patient. You can see that when the stimulation is on, the patient was able to stand up with partial support, with partial weight support. And the waveform on the right-hand side are the EMG signals recorded from his leg muscle to show the leg activities. It's really amazing and exciting for us to see these phenomenal results. And we indeed know how important it is for the patient to regain the lost functions. And as you might know, for most of the ACI researchers, the researchers place 1 or 2 electrodes on the cervical spine for the upper limb studies. And for the lower limb study, they might place another 1 or 2 electrodes on the lumbar sacral spinal cord to do some low limb or bladder researches. But as an engineer, we were then intrigued to know where and how does the current flow inside the body, and how does this relate to the effectiveness of neuromodulation after ACI. Our curiosity led us to explore more. So here is a realistic human model that incorporates skin, fat, muscle, nerve, spine, internal organs, and fat tissues. So we use this model to run electromagnetic stimulation. So as you can see here, the purple dot is a non-invasive electrode that we modeled. The electrode is placed on the back of the model. And also on the front side, we also place 2 return electrodes on the iliac crest. And then let's take a closer look. When the current is delivered through that single electrode, let's remove the skin, fat, and muscle layer and take a closer look. You can see that the current spreads on the spinal cord. And what does this mean? This means that, for example, when you place an electrode on, let's say, T11, you might be stimulated a much wider area or even off target. At the same time, we also worry about the safety of the non-invasive approach. Unfortunately, less current or minimal current, very minimal current flows to the heart. And how about cervical stimulation using a non-invasive electrode placed on the neck? And this is what what others have done a lot in most researches. We then developed a software that can demonstrate the electric current distribution inside the body during neuromodulations. So on the left-hand side of the model we built, the yellow circle here is the electrode placed on the C4 or C5 spine on sex spines, non-invasively. On the right-hand side is the current distribution when the current delivered through that yellow dot electrodes. And what surprised us is that for the cervical stimulation, you can see that most of the current flows to the nerve instead of focusing on the column. But also this depends on where you place the electrode because of the curvature of the cervical spine. That is a key factor. Let me play this again. It is also interesting that we found that the cervical stimulation, a lot of more current is focused on the ventral instead of the dorsal roots, even the stimulation delivered from the neck of the patient. Based on analysis, the inside observation, and our discussion with our collaborators, then we are asking these following questions. Is one electrode, two electrode stimulation good enough? And is the current delivered? Can the current deliver to the or is the current delivered to the desired spinal cord target or the biological tissue at the right timing, at the right location? Although we think it might be. And should the electrodes for stimulation be placed always on the same locations, even for patients with different injury levels? And we ask us the last question: how can we do to develop a product that can help the clinicians, researchers, and scientists, and even patients? And the last question led us to start our product development. Building upon our platform technology and taking variable inputs from clinicians, researchers, and patients, we developed XSTIM, exact stimulation, non-invasive neuromodulation system. And very critically, we also have the FDA engaged early on in our product development process and also get involved in our clinical study design. Their guidance is of extreme help to make sure that we are taking the right steps. And here is the picture of XSTIM. It's designed to be used for home use and in clinic use, and also as well as a tool for researchers to explore different avenues. The stimulation device is connected to a unique electrode array that can be placed on the body of the subject. The patient can hand the device, either hand or clip the device, either to a wheelchair. And we also have a versatile graphic user interface that's developed to be used by different users. And they can also be used for researchers who always have some innovative stimulation paradigms to explore. We'll share more information about our product in our future events. We then started our clinical study last year in October. So honestly speaking, it was very challenging to initiate a study during the pandemic. But fortunately, after overcoming some hurdles with our clinical PIs, now we are running our study at 3 US sites. I do want to show our gratitude to our clinical PI and collaborators, Dr. Sanko at Houston Methodist, Dr. Rebecca Martin at KKI, and Dr. Monica Perez at SI Lab for their help to support our clinical study and help us explore the potential of our product, and always provide us very, very valuable and insightful feedback. I also want to share some video with you. Here's a video of one of our patients before and after he finished the study. This is an HOA patient. So the patient went through 8 weeks of rehab and sham stimulation, and then another 8 weeks rehab with active stimulation. And in this video, the patient was performing a 9-hole peg test. You can see that he was able to move the pegs faster and be more precise after the treatment. All right. So Here's okay. So I want to also share another video with you. So this patient was doing the test to pour the water in a bottle into a cup. You can see that after the treatment, he can finish the test within just a few seconds. Compared to the video on the left-hand side, he has problem holding the bottle, and he will drop the bottle. All right. Okay, and then let me summarize my presentation today. Untitled is emerging company, despite the company is young, but we have developed advanced product and technologies, and we have a very, very strong clinical and scientific team. We are providing a new solution that has a potential to help the ACI patients recover their motor functions and beyond. Our product has a very good user-friendly interface for clinicians and patients, but also can be used as a versatile and powerful tool for researchers. We are looking for clinicians, patients, researchers, and other partners to work with us as a team to unlock the potential of our technologies and bring it to help the patients in need. Please feel free to reach out to me if you are interested in talking about it further, and also look forward to seeing you in future events, and please also pay attention to our progress update. Thank you.
All right. Well, thank you to the three of you for your presentations. Ikai, Keith, Uzma, very much appreciate you being here. Let's kind of jump right in. So I firstly want to kind of address maybe the obvious one, maybe kind of an obvious assumption. Keith and Uzma in particular, you know, some people might have listened to your presentation and thought, oh, wait a minute, are these two at odds in terms of their different strategies and approaches? How would you respond to that?
Well, I would have to say I agree with Keith 100%. I think that we are in the middle of learning about this. We've started a little bit and we have a long way to go. And, you know, for a patient who is, you know, struggling with acute spinal cord injury or chronic spinal cord injury, I think they have to have an understanding of, you know, what their options are, what the risks, benefits are. And as clinicians, it is our obligation to not give them false expectations, to sort of tell them what we know, what we don't know, and be reasonable in our explanation of risk benefit so that they can make an educated decision.
Yeah, I would say I would never want good to be the enemy of perfect, but I certainly want to make sure that we get to good. And by that, I mean that we have enough study of what's going on in whom and in what way that we can somewhat predictably prescribe this as a therapeutic intervention. Does not mean I would not try it if everything else has been tried and people might benefit from it. But folks need to know going in that this is experimental therapy, and their obligation is to be part of the data collection and learning curve for this understanding of the neurophysiology and this technology development of the devices and the equipment. And if everybody's willing to acknowledge risk and understand that there might not be personal benefit and is willing to contribute by participating in data collection, I'm certainly not against it. But I think there's some things we could do that would be a little bit more careful. I mentioned the trying transcutaneous stimulation to see if there's effect at all, and if that effect is at all predictable. It might be a good argument to do before you implanted a device. We certainly tried this with intrathecal baclofen and with pain stimulators. So there's a transient trial period after which a decision is made based on criteria as to whether full implantation should go ahead or not.
And Ikai, you in our talk just before we went live, you made a comment. We were also talking about just the need for disseminating good information. I mean, educating people about what this technology can and cannot do, who it can and cannot work. You know, all those questions are needing to be unpacked. You want to comment on that as well?
Yes. I think this technology has been around for decades, or I would say at least a decade. And since some of the companies are trying to, like us, we're trying to commercialize the product so that it can bring to the market to help the patients. So I think the community or us as a whole, we should provide fair and balanced perspective to both patients and clinicians to help them to have appropriately set up the expectation to know that what these therapies can do and cannot do, and also need to be clear about these therapies are just intended now to restore some important functions to improve their quality of life. It's not curative. It's not a cure. It's just at least for now, it's a treatment to help the patient improve the quality of life. And this kind of make our driving goal is to help patients go back to the life where they can establish a new normal that isn't completely limited by their injury.
So I have kind of relatedly another question. This term, and I think Uzma, you used it. I can't remember, Keith, if you used actually the word optimization. But it's a word that gets thrown around a lot, and I've heard kind of through the grapevine that funders, and particularly in peer review grants that use that word optimism, we're going to optimize, we're going to figure out how to measure and improve this technology. That in a grant, that's a kill word. That if you're going to optimize, then you're not going to get funded. You care to comment on that particular thing? I'm just curious.
Yeah, so I think the reason the word optimization gets used is initially there was a best recipe based on 1, 2, 3, a handful of patients or subjects in studies. And it became very clear that that wasn't going to work for everybody for all reasons. What works for autonomic dysfunction or for standing or for stepping or for hand function is not necessarily going to be the same even within one individual. And so the question then became, how much can you check the parameter space, stimulation frequency, intensity, timing, patterns, locations of stimulations, all these sorts of things. And it's with all the different degrees of freedom of the parameters, it gets very difficult and time consuming to do that. If you then recognize often in these grants, there's no description not only of how that will be done with any sort of a logical method, but then there's no measure of what the criteria by saying we've in fact optimized this is. Is it better EMG? Is it better robotics or better kinematics, better video, better feeling on the part of the patient? It's a little bit hard to know what those things are. I hope I got across that two things, that there's a lot of variability within the patient population. With regards to the effect of stimulation on stepping, if half the population is being affected one way and the other half in the other way, you kind of need to know which sphere that the patient's going to be in before you even start making assumptions about how it would be best to stimulate them. And then even within the behavior, I was pretty surprised when we found out that the effects on stance versus swing during stepping were more often than not opposite of each other. Nobody got augmented in both or inhibited in both. And so I think when we get to reaching and grasping, or if we get to bowel and bladder function, if we get to sexual function, there may be things that, to get one thing to go up, something else has to go down. And we just don't understand the underlying physiology quite enough yet. But I think with deliberate study, we can do this. Or if people are going to do this as experimental therapy, they can at least sign up to be party to ongoing investigations about what is the effect of changing these sorts of things, or studies that allow them to be better characterized so that we can know who responds like in group A and who responds like in group B.
And so is this, Uzma, is this something kind of that is almost makes a case for, there's a lot of people who would like to try this, and there's a reason you're suggesting, oh, well, let's try it. I've heard a lot of people say, if we can get more in humans and continue to study it, we're going to know more.
Yeah, my problem, at least for my patients, has been many patients come to me and they want to be part of a clinical trial, and I want them to be part of a clinical trial. So I've conducted trials at 2 different hospitals, and I've enrolled anyone that I can. And the problem is that we're limited in terms of numbers that we can enroll. And then we also, when you design a study, when you go to the FDA and you say, hey, I want to do a study, you have to have particular inclusion and exclusion criteria. And the problem is that because of the heterogeneity of this injury and the nature of people who have problems, it's very hard to design a study that's inclusive enough that everyone will be eligible. And so almost all of the patients that I've implanted off label, I've done because they aren't eligible. They don't meet the inclusion criteria. So either they weren't a trauma, they were, they were the result of demyelinating disease or an inflammatory process or infectious process, or, they coming from something else, or, they didn't meet criteria because they had partial function. So, for those patients, the options are very limited. It's unlikely that they'll be able to get involved in a clinical trial right now. Once they're implanted, we may have ongoing trials that we can put them in, and that's the hope. I think the ideal way to move forward would be the way the cancer community has, where literally almost every single patient who gets any type of cancer either gets an algorithm that has already been trialed as part of a clinical trial and is shown to be effective, or gets put into a clinical trial. And the cancer community has sufficient funding that they can do that. And spinal cord injury does not. And that's the tragedy of this research field, is that there's not enough capacity in the current trials to study everybody. And that's really not fair. It's not fair to the patients. It's not fair to their families. So somehow we have to do what we can to fill that gap. And we have to, I agree, we have to implant some people, knowing that they don't qualify for a current implantation trial, but then hope that they will qualify for a trial that will ameliorate some of their symptoms and make their function better.
Hey, Matthew, I'll just add that I've reviewed a fair number of grants for these kinds of studies, but very rarely and almost never has the inclusion criteria been, you have had an implant. We now want to ask these questions after the fact. And we're now tens to hundreds of people with implants. And I'm waiting to see a grant that says we're going to monitor the following parameters over time, or we're going to test alternate parameters, or we're going to study you as an individual more. And so if the people getting them are willing to look at themselves as essential to advancing and improving the technology and the therapeutic application, then we will go forward. But at this point, everybody's trying to figure out a way to get money to do the implants and to study them to show some sort of efficacy, as opposed to having the common denominator be, you got an implant, now we want to learn more from you now that you have an implant.
So Keith, we did submit a grant to the VA for that already once, and it didn't get funded on the first round. We'll be resubmitting it. So, excellent. And there was feedback
made this question about optimization.
They weren't happy about the fact that we included patients who had prior implantation. UK, go ahead. Sorry, I didn't mean to interrupt you.
Oh yeah, sorry about that. I lost my Bluetooth connection for a few seconds. So I want to add a few points about to address that the question about the optimization question that I made to ask before. So I think the better word from our perspective probably will be personalization. In our ongoing study, we do discover that there are some patients who need different electrode configuration. They need different stimulation target. They also need different stimulation parameters. And this kind of stuff make us wonder that personalization will be the better goal for each patient, because different patients have different injury. And this kind of why we kind of are building a very flexible and versatile platform. So that allows us to adapt our product and also adjust all the discoveries during our clinical study, so we can improve the effectiveness of stimulation and also provide a better therapy to the patients.
Okay. Listen, I want to pull in some questions on the chat board, and before I do, I also want to say we're probably not going to get to all of them. So for each of you, you can go on the chat board and reply to ones that either may have come to you or that you'd like to answer. I think folks would appreciate that. But let me roll up a couple, particularly from Anita, Peter, and even Nancy. Stimulator location, why that's important. Nuzma, I know you brought it up in your presentation, and in a sense, kind of tagging on to that, would more channels for the stimulation allow you to target more function? A question from Nancy. I'm kind of wrapping those together.
I think that more channels and smaller channels, so that you can more precisely localize a particular region, might potentially be beneficial. We do intraoperative physiologic testing when we place the device. So what I do is when I do the laminectomy and I put the device on the spinal cord, like in the epidural space, I stimulate each channel separately so that I can see what sort of impact it has on the testing of the lower extremities. And what we have found is that sometimes when you stimulate in one area, it causes movement in more than one area of the leg, which means that we're getting sort of a stimulation of a pathway, not just of a particular locus. And when you stimulate the whole pathway, then the whole pathway gets activated. And I think that's one of the challenges of this, because then, you know, if you get, for example, every flexor or every extensor all at the same time, that's not going to allow you to have the type of movement that you want to see. So smaller channels and more channels, I think, might potentially benefit our patients. And I wish there were some really easy way to break down the stimulation parameters such that you could figure out which ones correlate with which one, you know, and compare them from one patient to the next. And unfortunately, there's not. It's exactly like you guys said. It's very personalized, and we have found that. So when I put these devices, and I've put in 25, I think, at this point, and every single one of them is different. And, you know, what's interesting is in most patients, if you're directly centered on the spinal cord, you get symmetric conduction. And in some patients, you know, you actually have to be off center because the right side or the left side might be more or less injured than the other. And so it's different. You know, that makes it more challenging. And I think that, you know, direct personalization of each stimulator placement is the only way to do it right now.
Keith, how does that go? Oh, go ahead. I should see you're going to answer.
Well, I just would say that I bet it's really important in some physiological profiles, and I bet it's not important at all in others. And I say this because you can move the transcutaneous electrodes a couple of inches and still get the same reflexes and the same effects in some individuals and not so much in others. I also would point out it kind of may come down to how much you are doing the thinking for the nervous system versus letting the intraspinal circuitry do its work. So I tried to show you that the lumbar spinal cord in humans can, at least in some conditions, turn tonic input into phasic output, but unreliably and unpredictably so. So there are other strategies going forward where the thinking is being done by the interventionalists. So different sensory inputs are being stimulated at different times to probably more likely augment sensory feedback that would be happening during the behavior anyway. So I think it sort of depends on what the goal of the intervention is and how much control the interventionalist has or wants to have over the intrinsic circuitry that's organized within the spinal cord. So I think it sort of depends is the right answer. And I agree that personalization is a good term, but in all of medicine, we have categorization before we get to personalization. So if you have a bladder infection, we care whether it's a gram-negative rod or a gram-positive cocci, because the classes of antibiotics are very different for those two categories of drugs. But ultimately, your infection gets tested against a specific antibiotic, and you are either sensitive or not sensitive to that, and you get that. But even then, the dose might be too much or too little to generate either side effects or ineffective treatment based, let's say, on your body weight or something else. So in all of medicine, there's a level that is sort of more global, done by category. This would occur certainly in Uzma's example of cancer, but then all the way down to individual parameters when it comes to dose or frequency or exactly which chemotherapeutic agent is most effective in your specific physiology, in your specific tumor. So I think how much personalization, I'd hate to think that we had to do medicine from start to finish over and over and over and over again, and each individual never learning something about the population so that we could at least do shortcuts as people come in. I mean, therapists look at patients. I use the example of the weak hand in occupational therapy. If you show an occupational therapist a person with a cervical injury, immediately they have a different idea if that hand is flaccid and atrophied versus if it's very tightly spastic and got good muscle tone. They're going to take entirely different approaches to that physiological profile, even though the injury is the same. So I think we have to have the same sort of attitude when we go to spinal stimulation. What is this profile and how are we trying to influence it before we can start answering the questions? And I just don't think testing through the entire parameter space in everybody is going to be an effective route to learning this. So I think we may need to study some frequency, some on intensity, some on localization, some on pattern, some on spasticity, some on stepping, some on bladder function, et cetera, et cetera, et cetera.
Ikai, I see you nodding a lot, and it kind of lends itself to a question that I've been wanting to ask you, which is also raised by Sasha Rabchevsky. How is your device different? How is the approach of Anuvo different in the context of this conversation, even?
Okay. So I think the key difference in our kind of strategy is like I would say that we are more technology. I think we are more focused on technology and the therapy platform instead of just building a product. So we are trying to make a platform, as I mentioned before, very flexible, so that can adjust multiple therapies and disease states versus just build a single medical device. So that our goal is to make the platform adaptable, so that as we get more clarity about the responder, non-responders, or as we collect more insight about what therapies will help or benefit the patient most, we're able to include this and adjust these discoveries inside our platform. So that kind of makes us different from the approach that people are taking. We're kind of using what we know and guess what we don't know, and use the technology developed to incorporate that's what we don't know to explore those unknowns. Hopefully this is an okay explanation. And indeed, there are too many stuff we don't know. Yeah.
Okay. Let me ask, there's a question here from John, who's been putting up a number of comments and questions. This last one, why can't there be broad application of stimulation technology across the population to gather large amounts of subjective data to be shared, and then from that, targeted studies designed?
Oh, that would be an awesome thing.
Let's do it.
I think there is some consideration of cost and risk. But aside from those, I think the biggest hurdle for that specific activity is the metrics for success in academia and in medicine, that people compete for the money instead of working collaboratively. And it would take a funding mechanism that only gave you money if you worked with multiple other centers to do the large kinds of studies, and maybe even specialized out that at one institution that focuses on this aspect and this other institution that focuses on that aspect, but all the data is going into a common database for analysis by the entire group.
So the cancer groups have done that. They build consortiums and the consortiums work together. So it's possible to do. The PEDS neurosurgery groups have done that. They've got the hydrocephalus associations. They've got Chiari foundations. They have large organizations that band together and pool the data. So I don't think it's purely an ego thing. I know that most of my colleagues that I work with are more than happy to work with others if it's in the mutual interest of patients.
Well, I was going to mention, it's a good segue, I think, to mention, which I wanted to touch on. On Monday we talked about our translational work group, or not, on a first day session, and then the second day session we have an activity-based therapy work group. For the last few years, we also have been facilitating a neuromodulation work group, of which Keith has been a part, and a number of other folks. And I think it lends itself to what you're talking about. One of the actions that this group decided to focus on, and these are all scientists and clinicians doing stimulation in human beings, was to create a kind of consensus around, okay, who are the people that are getting these stimulators implanted? What's the patient profile? How are we measuring effect? What kind of tools are we using to do that? Because there didn't seem to be real consensus around that. And then even from that, okay, well, what are you doing? What are you stimulating? And how are you doing it? And this group has put together a survey. It's finally just been completed. We should be going out soon once we get approval from the Mayo Clinic who's housing it. And that's kind of a step in that direction in my mind. Would you, Keith, I know you've been involved in this pretty deeply. Would you agree with that?
Yeah, the first step was to make sure we're comparing apples to apples across studies, because if we're not recording the same information or collecting the same thing, there are a bunch of one-offs that have a hard time going together. So establishing at least common denominators for what information will be collected and documented as to what the interventions are and how they're being followed. Hopefully this will lead to a collaborative environment and attitude that since we're all getting the same kind of information, it can go into a common database, and then we can start doing analysis across centers because we have the same data points to compare between centers.
Right. Well, listen, I think we got to wrap it up. But and again, I just want to mention there's a lot of questions up there. So I think folks would be really appreciative if you were able to reply to the ones that seem relevant to you. I think it'd be great. And there's Ikai, especially, there's a few questions directly at Anuvo, questions about Anuvo. So I think that'd be fantastic. Again, I really appreciate, I think Sasha was one who commented how much the clinical work that you do all do is so underappreciated. Thank you. So I think that's, I want to give you also thank you like that. Very much appreciate your presentations and giving time to us today. And again, like everybody, I would love to have you come back to the breakouts later after the symposium is over at 3 o'clock to join whoever shows up.
Thank you, Matt, for organizing this. You've done a great job.
Thanks so much.
Yeah, thanks, guys.
Okay, so we are back. Sorry for the little delay. One of our panelists is having a little trouble getting in, so we'll pull him in once he's here. I want to introduce you to Tracy Fernandez. Tracy's one of our board members, also has spinal cord, should I say SD?
That's fine.
And what we really wanted to do with this, and hopefully Rob will come on to join us soon. I'll introduce you to Rob as well. Rob has a spinal cord injury and actually was a participant in the E-STAND trial that Uzma was discussing in her presentation. And so you just heard these 3 presentations. We talked about some of the research and some of the science. Now we wanted to talk with people who've experienced firsthand spinal stimulation. And so that's what this is about. So Tracy, why don't you tell us a little bit about that?
Okay, so I was fortunate enough, well, a little bit background on myself. I have a T4 level considered injury, I guess. It's from transverse myelitis that was a result of an illness, transverse myelitis, back in 2008. So roughly 13 years ago. Because, and I think one of the previous presenters mentioned this, because it's transverse myelitis, it does greatly inhibit some of the trials that you can be a part of. It's not a traumatic injury. And so I've always been interested in clinical trials and being part of that process. You know, what can I help do? Not just for myself, but, you know, for the greater good of the community. And have been always looking at that aspect of it. And through being part of U2FP and also being here in the Chicago area and kind of looped in with Shirley Ryan, I found out about a clinical trial that was going on up at Shirley Ryan that involved really a 2-pronged approach. Where it was using both the transcutaneous spinal cord stimulation that you just heard about, as well as something called transcranial magnetic stimulation. And that is done where they basically take, magnetic stimulators over your primary motor cortex, and they use that to help activate the cortical spinal tract and the neurons. So you're basically stimming almost above and hitting the spinal cord simultaneously from 2 different areas, from above it and kind of below the area of injury. So that's what I was involved in out at Shirley Ryan. It involved 40 sessions. So really for mine, it was 4 days a week over the course of 10 weeks. So it's quite a commitment. And you're actually at the location for about 3 hours a day, because you're being stimulated for me, it was 30 minutes during the session that I was in. So the stimulation happens for 30 minutes. So it's 5 seconds of stimulation and then 30 seconds off, and then 5 seconds on and then 30 seconds off. And then that's followed by, I think, a little different than what Rob will tell you about, where I don't believe he had PT afterwards. The mine was followed by about an hour and a half of physical therapy. And that was primarily involved some bicycling to see, if you had the capability of powering the bike yourself, that's great. If you didn't, the bike would assist you, and then followed primarily doing overground walking. So you're in a harness, you put on AFOs, and then someone helps assist you move your legs forward. So that was what the clinical trial itself was about.
Okay. Hey, Rob, glad you could make it.
I just had one of those technical difficulties type situations. Sorry about that.
So first off, why don't you just introduce yourself and, like just Tracy did, explain you were part of the eSTAN trial. So just give us a quick snapshot of that.
Yeah, absolutely. I'm Rob Kozarek. I am a T4, T5 complete para. I was implanted with the eSTAN trial in November of 2019. So the main thing about this trial is that it kind of follows along a very similar path to previous research where you're implanted with a spinal stimulator within the that can be used to generate movement. There's also an autonomic response that they're looking for. What's different about this particular study is that with the current status is that you're implanted and there is quite a bit of intensive rehab that's associated with it. Through the eSTAN trial, they removed that aspect of it, the physical therapy aspect of it. So you're just given exercises to do with your stimulator to see if it can work in a regular everyday modality, and to see if there's a response to it. The overall response they've gotten so far has been really great. For me, I was really focused on the autonomic responses, so bowel, bladder, sexual function, that sort of thing, and I got really great responses out of that. The trial is broken up into 13 separate visits with the team. Through those visits, you're continually refining your settings and the ways that the stimulators that is stimulating your spinal cord to really hone in on settings that really work for you. The overall way that it works is through an app. You can actually Bluetooth connect it to your battery pack, which is surgically implanted. And you can kind of pick and choose settings that work for you. I use it every day, and I have gotten some really great responses out of that.
Yeah, so let me a couple things I want to touch on. One is that I'm glad you pointed out that I don't know if this was the very first, but certainly a unique element of the eSTAN trial was untethering rehab. All this, like people have seen the treadmill walking and all this aggressive rehab, both pre and post, right? Pre-implantation and post. This was not a part of the game with the eSTAN trial. And then the second unique element was this, like you go home and you get to try setting one and see how it works we think this will help with movement, or we think this will help with autonomic bladder function or bowel function. So to the degree that you're willing, I don't want to make you, but like when you say you had really good, like what came from it that you had?
Yeah, what I'm happy with particularly is, previously with bowel and bladder specifically, I had some pretty intense bladder spasms that would cause me to look at medicines to kind of cut that down. I've completely reduced that to nothing. I don't take medicine anymore for that, any kind of medication. So that's under control, which has been great. and then bowel program and control, or bowel programming and control, I've seen huge improvements where, you know, bowel programs can take anywhere from 45 minutes to an hour. Now it takes about 20 minutes. So if you kind of extrapolate that over the course of a week, you're saving hours and hours of time and pain and anguish. So that has really shown some really great responses to that. Another kind of interesting thing that they're still kind of looking at too, and this is actually kind of goes into a really important aspect of this trial. One thing that I've noticed is that I'm an athlete and I bike, I'm constantly looking to stay busy. When I have the stimulator on for autonomic response, I've noticed that I can exercise longer and harder. Now, what's important here, though, to show is that is actually blood pressure control. And so that's something that's huge for people in the spinal cord injury. I don't have like a lot of issues with blood pressure, but I know people that have in the study, and they've seen a huge response for that. And that's something that relates to a lot of different things, you know, not only comorbidities or anything like that, but also you're looking at temperature control and that sort of thing.
Cool. Tracy, what about you? What was your experience in that study?
Yeah, so I think for myself, there was small improvements with the transcutaneous and the magnetic cranial. The people in the trial who definitely got the biggest improvements and the best benefits were the people who were the most incomplete. So Asia, C, and D. Myself, they do a multitude of testing at, you know, the beginning at 20 visits and then again at 40 visits. And so while I had, you know, every muscle group did show improvements, but if you're going from 0 to maybe, you know, a 10% improvement, you can start to detect little flutterings. That doesn't mean that you're able to kick out your leg or that you're able to stand or lock out your legs or anything like that. Now, individuals who had, you know, better movement than myself did see, you know, much greater improvements. So, but myself and others that were similarly, you know, I can move my toes on my leg, so I do have some movement and I can bring my foot up on my left. But it wasn't like I was able to kick my foot out or anything like that. But I did see anywhere from a 10 to 30% muscle activation improvement, but nothing I would say significant enough that would cause any type of, you know, functional improvement.
Mm-hmm. So, and I think it probably should be known if people haven't read your bio that you run a activity-based therapy center outside of Chicago. So I'm assuming you utilize that to the best of your ability, right? I want to we only have a couple minutes, so I do want to, there's a couple questions here I'd like to kind of roll up. But one from Chris. Rob, can you give some more specifics? What type of stimulation and where is your, I mean, it's epidural stimulation. Where is it placed? Mm-hmm. And use different stimulation parameters for different functions.
Yeah, absolutely. Absolutely. So where the paddle is actually placed is in the epidural space at T11. So that's below my injury, but that's a standard for this particular research group. As you all know, research programs need to have very standard ways and very standard parameters. That being said, the way that the actual app is set up and the way that the study is set up is that you get 10 volitional settings and 5 autonomic settings. The 10 volitional settings will target different areas and different spaces to allow you to do numerous different things. The biggest response that I have, and I can do this very consistently, is kick out my legs when it's turned on and turned up. So I can kick out my legs. I can lift my knees up depending on the setting. You can also they also have settings to help focus on ankle movement and movement nature. And then for the autonomic settings, they do have ones that target more more along the lines of looking at blood pressure. They do have ones that work better for bowel. They do have ones that work better for bladder. So it really, the main hindrance I have here is that a lot of the information was shielded from me since I was a participant. But the information is out there in white pages. Dr. Darrow has been putting out white pages on this or white papers on this for years now. So the responses that they have and the results that they've generated are promising.
And also to both of you, maybe starting with you, Tracy, there's a number of questions about trunk control. Any experience, any changes there for either of you?
I think when it's on, with the transcutaneous, because you're only using it for me, for example, it's only on for 30 minutes. And you're seated at that time. But, I think from a trunk perspective, yes, you do see some benefits to that. But again, the stimulators, it's placed, the higher level stimulator is placed right kind of below your neck. And then the lower one is obviously, to the right around T10, T11. So you're going to naturally get some of that kind of bringing you back. Right. Just like what if it's any kind of stimulator.
Rob?
Yeah, I have my my stimulator on throughout the entire day. And a lot of the residual effects of that is that you do have a lot more, it does affect your trunk in some aspect. Some settings, of course, affect it more profoundly. But overall, there's an underlying kind of undercurrent of stimulation going on at any given time, and that results in a really solid base for your for trunk control. So I've noticed quite a bit of trunk control. I can do. You can almost do sit-ups with this thing sometimes too. It can be that strong depending on how strong you want to turn it up. The one caveat there, of course, is if you have it up constantly, there's a pain aspect to it, and there's also a possibility of you draining your battery faster.
Well, listen, we got to wrap it up. This went so fast. I wish we had more time to talk. But just like I've been telling other panelists, there are questions that we didn't get to. And if you guys would be willing to reply to folks that are questions directed at you, that'd be fantastic.
And I'm hosting the session at 3.
Oh, cool. Oh, excellent. Yeah. So if you want to ask Tracy questions, or Rob, if you're able to make it at the breakout session, they'll be there so you can ask them questions directly. So thank you so much, you two. Appreciate it. And also, just kind of looping this back together, I think, the ESTAN trial was actually one of the first things in the Minnesota State program when we passed our bill. It was the very first thing that we funded. And I think in my recollection, being on the advisory council, I remember being kind of intrigued by this untethering of rehab and stimulation, just to see, okay, well, let's see what happens without that. And then also this, kind of applying, an A-B choice, like, is it better, like when you get your glasses done, basically, left and right, like, is it A or B? A or B? And that's, in a sense, what the studies that Rob has participated in was doing. And I remember that and thinking, and it's cool to see it all this time later come to the kind of fruition it has. So really appreciate your time. Thanks for being here. Now we're going to switch gears again and talk with folks at ONWARD.
Absolutely. Have a good one, guys.
Bye.
I go by G. I am professor at the Swiss Federal Institute of Technology.
And I'm Jocelyn Bloch, and I'm a neurosurgeon in the University Hospital of Lausanne.
And together we are co-director of NeuroRestor. NeuroRestor is an academic center between our two institutions that really aim to develop treatment involving neurosurgical intervention with various neurological disorders, including spinal cord injury. And because we really are meaning to develop treatment that can be used throughout the world by everyone, we have a very strong collaboration with companies, especially ONWARD. You can see how our logo fit together because our philosophy is really to go all the way from an in-depth understanding of the underlying science. We've worked in preclinical model, mice and rats, amplify our discovery, with work in non-human primates, and eventually proof of concept in human that we can pass on to ONWARD in order to develop real-life interventions. And we're going to show you today 2 examples of this translational path. And we will start with the mobility. We've been working for 20 years in developing what we like to call the Swiss watch precision in the way we stimulate the spinal cord to restore walking. Because the way we do it is very different from others. We apply bursts of stimulation at different locations with a timing that coincides with the ongoing movement. The underlying idea is to reproduce the way the spinal cord is activated naturally by the brain during the activity that is targeted, in this type, walking. So this is a biomimetic stimulation. You can see here David, for example, stimulated, he can walk, you stop the stimulation, he's paralyzed, turn it back on, and he can resume his stepping movements. This was developed because of all the understanding of the underlying science, which started with very precise modeling, so we can really visualize the electrical fields that are induced by the stimulation of the spinal cord. And what we found, combining this with anatomical models, is that the stimulation does not penetrate the spinal cord. It flows on the side in the cerebrospinal fluid, where it will depolarize the neural structure with less resistance to current. And these structures are the large diameter afferents, the sensory fibers that are connected to receptors that are located in your muscles. So the dorsal roots are the spinal cord gateways to very specific neurons, which we also discovered using the more precise approach of single-cell RNA sequencing. We found that this one little population in the middle of the spinal cord is directly activated by this large diameter afferent and also by residual pathway from the brain, especially the reticulospinal pathway that is so important to control walking and leg movement. In turn, this neuron projects to 80% of the cell in the ventral region of your spinal cord, the region that produces movement. So we have understood at a high degree of precision the circuits that enable the stimulation to restore volitional control over the movement activity of paralyzed leg. And that was very important because then we could leverage this understanding to develop purposed technology that really leverage this understanding to translate this approach in humans. Right, Jocelyn?
Mm-hmm. So how does it work in the human being? So you understand from what G said that precision is extremely important and each individual is different. So we had to understand the anatomy of each participant to modelize the spinal cord and to build a specific lead electrodes that are able to stimulate very precisely each pool of motor neurons to activate the leg muscles. And the other important thing, we had to link it to a stimulator, a neurostimulator that was able also to give these stimulations patterns that is not available commercially yet, but ONWARD is working on it. So we implanted this lead epidurally over the spinal cord that is controlling the leg movements, and then the participant could step. They could engage into training, and that's an extremely important thing for them, is to be able to train regularly and for a long time. And not only one could make it, but the 9 participants included in our study were able to step and to train. You see that 6 of them are incomplete, the last 3 of them are complete, all of them could do these steps. But not only steps, since the lead we built was longer, we could also reach the roots that are able to control the trunk. And you see that these movements that are very difficult normally in people who have a lesion highly located are much easier with the stimulation. So this training, suddenly, after a few weeks, we saw that the participants regained a mobility that they had lost for many months and years without stimulation. You see here this completely left plegic leg, after 2 months of stimulation and training, regained movements. not only when the patient is sitting, but also when he tries to walk between two parallel bars. So for us, that was really important to see this plasticity, these changes after a few months, years of paralysis.
Plasticity, yes or no? Well, we have recovery of a neurological function without the stimulation, indeed suggesting that there is neuroplasticity changes. But is that true? And you know, everybody talked about it in the panels also, I'm sure. Like, what is happening? How is it possible that people improve their function? Well, we have developed a model that allows us, when we have this type of question, to go back to preclinical models and answer this question with the highest level of precision. In this case, we model this recovery in mice, and to evaluate the reorganization of the neuronal pathways, we rendered the brain and spinal cord completely transparent. This means that we can now convey a survey of the neuronal pathway without cutting, without any bias. You can see now labeled with viral technology, like gene therapy in a way, pathway coming from the motor cortex in blue, from the reticulospinal formation in white. I told you this is the fiber that connects to this neuron directly activated by the stimulation. And now you can see in 3D how this pathway are interrupted, but some fiber escape this severe contusion that leads to paralysis, and the spared fibers will sprout in the gray matter onto the key neuronal population activated by the stimulation, exactly where we stimulate. So it was a proof, at least in preclinical model, that this intervention promotes the growth of new nerve connection below the spinal cord injury. To enable, I mean, you have seen David take a few steps. It's not a cure, clearly, by far, but the stimulation, improve recovery, but also could support movement. So all our participants were frustrated to not be able to use the stimulation outside the laboratory environment. So to enable a proof of concept, can we use the stimulation spatiotemporal outdoor? We developed an engineering framework. So a connected watch that you can trigger manually or with your voice that enable to trigger the stimulation, select activity-dependent stimulation sequences. So for walking, standing, or other activities that I will show you. And you can trigger this activity, synchronize them with a movement using either ergonomic clickers on assistive devices, or for example, wearable sensors that can be either on the feet or on the device, etc. And using this technology, not only individuals with incomplete spinal cord injury, but also with complete spinal cord injury, have been able to take steps with the help of the ambulator outdoor, outside the laboratory environment. And not only they could regain mobility, but they can also perform other activity like recreational. For example, this is an individual, same thing with sensory and motor score 0, so complete sensory motor paralysis, with the stimulation able to do boxing. This is another one doing cycling, and in this case, the leg really produce movement. And if, for example, if you want to go up a hill, we stimulate more and more and more, so it really helps and give a really good sensation that the whole body is involved in the cycling activity. And as Jocelyn showed you, because we can now target the trunk muscle, we can also support activity requiring trunk stability. For example, paddling on the lake, which require a lot of trunk stability. So this is what we have been developing, proof of concept in 9 individuals to support mobility. But with this platform that we have developed, we have this work going all the way from mice to humans. We ask, can we now leverage our understanding, our technology, in order to target other neurological functions, which are important for people with spinal cord injury, because we don't have to explain you that there are many, many functions that are affected beyond the movement of the leg.
Yeah, one of them is hypotension, orthostatic hypotension, especially quadriplegic patients. Probably some of you know about it. It's very difficult in the morning when you start sitting in your wheelchair, feeling comfortable because your blood pressure is going down and it takes a while to be normalized again.
And others reported already that when you stimulate the spinal cord, there is an increase in blood pressure. So it was like an empirical observation. So we ask ourselves, like, can we leverage our technologies to understand the underlying mechanism and then exploit this understanding to optimize technology to target this function? So what we have been able to show, it was a really nice collaboration also with Aaron Phillips in Canada, that the stimulation activates the neuronal population that I showed you before, that this population actually connect to sympathetic preganglionic neurons, which then in turn activate neurons in the sympathetic ganglia, which lead to the vasoconstriction of arteries. So it's a reasonably simple circuit that you can target in order to increase blood pressure. So we mapped this very precisely, understood where these circuits are really located, which is not where others have stimulated, but it's in the low thoracic spinal cord. And we then designed this precise electrode array that target the dorsal roots, modulating the sympathetic circuitry. And in this case, we first did in rats and then in non-human primates. You can see here this low blood pressure during the orthostatic challenge. And when we turn on the stimulation, the blood pressure goes up. Turn it off, it goes down. Up, down, up, down. So we have very precise real-time control over the blood pressure. We did it in rats, in non-human primates. And when Jocelyn was implanting these monkeys, she thought, okay, it's time to go to humans.
So humans, it was exactly the same. So we localized the group of neurons that were able, this sweet spot to regulate the blood pressure. We did it in 2 participants, quadriplegic participants suffering from this hypertension. We again implanted the lead epidurally over this very defined zone. And we did not only see that we could really manage the blood pressure when they were laying down, but for us, it was very important to challenge the system when they were doing a tilt test, meaning that they were verticalized. And normally, as you saw in the previous slide, that in a few seconds, you assist to a blood pressure drop. With the stimulation on, you can keep a normal pressure for a very long time. And that makes a huge difference in terms of comfort.
So at this stage, we have done like 100 rats, monkeys, and 2 humans, as Justin said. So we feel confident with a treatment that is simpler to apply than to restore mobility that require rehabilitation. It's a bit more complex. This strategy, we feel, is ready right now to be delivered as a commonly available treatment throughout the world. And for this, clearly, it is not an academic endeavor anymore. We need a collaboration with a company. This is why we work with ONWARD. And this is why today, Dave Morver is with us in the panel to tell you about the vision of ONWARD Medical in order to develop actually a range of technology based on epidural electrical stimulation and the spatiotemporal stimulation that we developed, and make this a technology available for all the people in need.
So we're rounding the bend towards the finish line, and we are so grateful to have Gregoire, Jocelyn, and Dave from ONWARD and NeuroRestore. Thank you, Gregoire and Jocelyn, for the presentation. Welcome.
Thank you.
So, and we'll get to, there's plenty of questions. We'll get to that. I want to start with this. So even kind of the way we were thinking, there was just almost an hour of discussion about, actually more than that, discussion of spinal stimulation, and with a couple of folks who've participated. You represent a company taking this technology to the market. You are attempting to answer the questions that were raised in the previous hour. So tell us, answer some of those questions for us. Start with whichever one you like. And for instance, I'll start with this to kick you off. You both use the term purpose-driven technology. and I'm thinking, oh, as opposed to a repurposed driven technology. Would that be accurate?
Well, if I may, why don't we start, Matthew, by just talking a bit about the company and just establish the foundation. And then I'd be very thrilled to answer your question about what purpose-built technology is and how that differs from maybe the pain management devices that have been used in the previous and current trials.
Go ahead.
Sure. So ONWARD is a medical device company, and our purpose, our raison d'etre, is to develop therapies to help people with spinal cord injury. We have two different technology platforms. We have a transcutaneous and an implantable. We have 3 breakthrough device designations covering each of the 3 indications in our current pipeline. So restoration of hand and arm function using the transcutaneous, restoration of normal blood pressure and trunk control, and the ability to walk and stand using our implantable system. Those are called ARC-EX and ARC-IM respectively. All of that is protected by over 290 issued or pending patents worldwide, many of them licensed from the leading neuroscience research institutions around the world, such as EPFL, Caltech, UCLA, University of Minnesota, University of Calgary, University of Louisville. We have all of that at our disposal. And we have a management team as well that's very seasoned. Most of us came from the big med tech companies, and we're now deploying all of our passion and energy toward ONWARD and this spinal cord injury community. One example, John Murphy, our Chief Technology Officer, he actually ran all of neuromodulation R&D at St. Jude Medical, now Abbott and LivaNova, and he spent a good part of his career with very significant roles at Medtronic. So we have the team, the intellectual property, the technology platforms, the partnerships, and so forth. And we're working very hard to scale and commercialize these for the benefit of everybody on this call. And in fact, we already have 1 pivotal trial underway for the transcutaneous called Uplift, where we're enrolling 65 patients at up to 15 sites worldwide. We're beginning discussions about pulling together the group of investigators who will participate in our implantable pivotal trial for blood pressure and trunk control. In fact, there's an investigator meeting planned for the next month or so. So we have a lot of momentum here. I think most importantly is we've raised a considerable amount of capital to finance all of this. So this is why we now have, I think, the infrastructure and the capability to make a difference and to answer many of the questions that were raised earlier around using devices off-label, around programmability, around optimizing patient care after the implant, around reimbursement. All of these things we can talk about today. So thank you for that introduction, but I think it sets the table somewhat for the group.
Sure. Gregoire, Jocelyn, you want to talk about that purpose-built technology?
Yeah, what it's maybe important to stress is that you said like you represent ONWARD, but Justin and I really represent Neurorester, which is an academic center, really like having synergy between engineering and medical expertise, you know, in order to really understand deeply the science, have proof of concept that we can coordinate very tightly with ONWARD. You know, this synergy is very unique, and it's enabled to go very rapidly toward translation with the more precise understanding of the underlying science, and that every technology developed along the way, as you said, is purpose-made, meaning developed based on the understanding of the underlying science and really optimized for the application. I think this model is very unique, and we are very proud to have been able to develop it, because this is what you need in order to develop these innovative devices.
So help people understand regarding that specific thing, what is different about this new implantable device, for instance, from what's being used? You know, what's the difference in material? What's the difference in number of electrodes and the panel, the placement, and even the understanding of what parameters you're using and how you're using them? You alluded to that somewhat in your presentation. So Jocelyn or Gregoire, what's the difference?
Why don't I just take that at the start, and then maybe we can use a specific example. So really, Matthew, I think the era of using repurposed pain management devices is over. You know, we have developed an implantable platform that is purpose-built for spinal cord injury applications. That means there's a lot of flexibility around the stimulation parameters, the ability to stimulate through individual electrodes with specific frequencies. There's a lot of flexibility there. The system is designed to work in closed loop, so it can harvest a lot of data, wireless data from external sensors, and use that to inform programming parameters in real time.
External wearable sensors, you mean?
Could be wearable, could be implantable for blood pressure, for bowel, and so forth. We have our own waveforms. So we have our own ways of stimulating. We have wireless recharging. We can assimilate a lot of data with very low power consumption. All of these things have been engineered into the implantable platform. We also have our own family of leads that have been purpose-built for these indications, you know, not using these pain management leads. So Gregor and Jocelyn can tell you how, working with them, we have developed leads that can capture, you know, the right spinal cord anatomy, so that we're stimulating the right muscles and we're triggering the right autonomic functions. We're not inadvertently recruiting adjacent muscles or causing spasm or so forth. You know, all of this is so critical, so that the therapies themselves are repeatable, predictable, simple enough so that they can be used in any hospital or any clinic, and therefore spinal cord injury patients or people with spinal cord injury throughout the world can benefit from them, not just at these very, very specialized centers. So G and Jocelyn, maybe you can give an example of how we develop the leads with you. That might help as a good example.
Jocelyn, as a neurosurgeon, should explain.
So I think from a deep understanding of how it works, and as it was previously said, there is of course a difference of anatomy of each individual, and as it was also exposed previously, individuals are very important to take into account, but we understood how to see the anatomy of each and how to take into consideration the different routes in order to design new leads, not only at the lumbar level, but also at the thoracic and cervical, taking into account the anatomy of the different routes in order to be more specific, and then to apply the right stimulation to replicate actually the therapy. Maybe, Grégoire, you can add more about the different stimulation pattern we used.
So the key is that it starts for us with an understanding what we like to call the spinal cord gateways. The spinal cord gateways are the specific neural structure that you recruit to modulate the spinal cord. And those are the large diameter afferent fibers. And when you have understood the neural structure that you stimulate, you can run computational models that will tell you where you should put the electrode in order to be as specific as possible. And then, because you know that different regions of the spinal cord, let's say simply, will respond with different, for example, stimulation frequency, waveforms, you can capture all this location, timing, and feature stimulation in what they've called the waveforms, in order to be incredibly specific in the way you dialogue with the spinal cord. Dialogue meaning to activate motor function or blood pressure modulation system, even what we are seeing currently, the bladder function, and I hope soon the cervical stimulation for upper limb movement and recovery. So this type of very precise approach to the stimulation of the spinal cord based on hardware and software will enable to have a completely, it's not that it's better than what is being done with this stimulator for pain. It's really a radically new way to stimulate the spinal cord and to restore neurological function.
And I think, Matthew, the presence of let's say a significant industry player here is going to make a big difference because we will undertake the investments and initiatives required to get regulatory approval so these devices can be used on label, not off label. We will work with with CMS and the VA and the private payers to make sure, first of all, to recognize and identify whether existing codes can be used, but if not, to push for and assure reimbursement is in place so these therapies are covered and paid for. We expect to put to field and deploy a field organization with field engineers that can support the implants, make sure that the lead placement is correct, make sure that the programming is optimized during the procedure, but then also assist the spinal cord injury clinics with any ongoing programming adjustments that need to be made over time as the person regains function and musculature and so forth. We can also, again, addressing one of the issues that arose earlier, facilitate research and research collaboration. We have obviously a very deep relationship with NeuroRestore in Lausanne, but we're already working with 10 plus centers on the Uplift trial. And we're going to add 10 plus centers for our implantable pivotal trial. But I would say there are two other things that we've done. We have developed a layer we're calling ResearchGate now. We're probably going to change the name, but it allows researchers around the world, if they want to use our implantable system to experiment with new indications or therapies, they have a way to do that. And they have tremendous flexibility in how they can use our system to try new things. And we also, with our US organization, which is rapidly growing, we've put in place resources for investigator-initiated research. So we can support you with technology, with protocol development, with the conduct of the clinical trials, and even with the preparation of manuscripts. So we really want to play the role that big medtech traditionally plays in some of the well-developed, mature fields, and bring that those resources and that expertise to the SCI community as well.
So you're answering some of the questions that are popping up on the chat.
Oh, okay, we don't get to see those.
Yeah, that's good that you're addressing some. I think in many ways you just answered John Chernesky's question, but let me dig into that a little bit because there's some other questions, and I'll try to roll them all up. So there are a number of questions that I think get at this sort of target product profile, like the profile of the consumer base. And so in particular, there's some questions about incomplete and complete injuries, and questions about, well, so the gentleman in the video who is taking steps, is it Daniel? Is that his name?
David.
Sorry, David. So there's some questions about David and what his functionality was prior to stimulation and post-stimulation. And then also in terms of the trial, a couple folks asked about, again, as it relates to incomplete and complete injuries. So if you could tell us what is your thinking about the design of the trials going forward? And in particular, what do people with more complete injuries, what can they expect from this technology as opposed to maybe Asia Cs and Ds, but Asia As?
Sure. I think so. First, as from a company perspective, we want to facilitate research that allows as much learning as possible. But when it comes to designing a pivotal trial, which is expected to result in FDA approval or CE mark and authorization to release them, we want a trial that's going to succeed. There's a balance between architecting that for a broad enough population so that there's commercial viability, but a limited enough inclusion criteria so the odds of success are fairly high.
And therein lies the decision to focus on blood pressure.
No, because our current clinical trial is actually for upper extremities, and we're enrolling Asia B through D. That doesn't mean that we can't, that the therapy, the transcutaneous therapy won't work on Asia A, but the trial includes B through D. Our belief is that it's quite important for us to get a device out there, FDA approved, build a business around that, so that there's something out there that investigators can use much more easily to explore new indications, expand populations, and so on. I mean, that's the step you need to take. Get the thing out there and use so people aren't having to rely on experimental technology, and they're not trying to, they don't have to go through the regulatory burdens. So that's sort of the way we think about it. And it's the same thing with the implantable. We want to start with blood pressure and trunk control because, frankly, there's a larger population of people with spinal cord injury that would benefit from that. There's really very little, if any, rehabilitation required. Based on the Nature paper in January, you can see this hemodynamic hotspot has been identified, and we stimulate in that lower thoracic area in a very specific way, and it's getting good results, all to be proven, of course, in the pivotal trial. But we decided to start with that because it can benefit more people.
So there's a question, I mean, directly in response to that from Krista, kind of piggybacking off mine. Why not consider the zone of partial preservation simply from the publication in 2019 to target in more complete injuries? Is maybe that's a question, Jocelyn Gregoire.
Is this the 2018 Nature publication describing the walking? Yeah, I think there might be a publication coming that answers that question, perhaps.
Okay.
Mm-hmm. Jocelyn, you want to take this question, maybe?
Don't disclose anything that's not been published yet, please. We're a public company now, Matthew, so we just need to be
sure, I understood.
We need to be a bit careful about these things now.
Yeah, I don't want you to disclose anything you can't, of course.
Mm-hmm.
I think that in the presentation, some things were already said.
Okay. I'm not sure I understand the question exactly. Are we going to try to help people who are Asia A walk again? Is that the question?
Oh, I don't think that's the question. I think it was related to that decision regarding your pivotal trial and the focus. Obviously, you do want a trial that works. Mm-hmm. But I think the question directed at, in Asia A, I'm assuming the questioner is asking, there are some preserved pathways, and could you expect to target them, and why not include them?
Okay, I understand. So yeah, indeed, we think that there is promise for the therapy in Asia A, but we made the decision not to include them in this pivotal trial because we want, obviously, we're trying to balance a broad population with the success of the trial, the odds of success of the trial. So pragmatism. Go ahead, G.
And this is Dave talking like as the company. I mean, Jocelyn and myself, we are clearly associated with ONWARD, but also like academic researcher with more freedom, maybe investigating. And from our experience in hundreds of rats and over 10 individuals with spinal cord injury, I mean, we know that there is a correlation between the severity of the injury and how much you can expect as a neurological recovery, meaning recovery without the stimulation. However, we also know that every single spinal cord we have been stimulating, would it be incomplete or complete, in rats, monkey, pigs, or human, have been responding to the exact same way to the stimulation. It's highly predictable, the effect of the stimulation. And in that sense, we should really now have this purpose-built technology, move them to a large number of people to really understand what we can bring with different individuals, because we will have different outcomes with different type of injuries. That's my take to the perspective, the question that was asked.
So one of the things I wanted to ask is sort of relatedly is, in the conversation previously, particularly around two things, this idea of, and certainly sort of trying to burst the frame of either or. There are these studies going on currently, right? And there's learning from that, whether it's at Louisville or Mayo or University of Minnesota or wherever it's, VA, and even USMA's strategy to, go clinically, right, with the given technology as it is. And if I'm assuming you may not have heard yesterday, Florian Sulzbacher from BlackRock Microsystem, a device company that's been around for a decade or more with the Utah array, and he made a particular comment about for him and their company, this sort of moral and ethical challenge of not needing a perfect technology to roll out. There are people who want function now. And so my question is, what can you learn from the ongoing studies? How does that feed into this question around the target profile, and even more pointedly, this kind of data set from the patient profile? Who is being measured? What effects are being measured? How are they being measured? That we've been sort of working to develop over the last couple of years. How does all this feed into what you are all doing?
Yeah, well, thank you. I think that's
feedback. I know you're a private company, so there are some things that you.
No, well, why don't I take the clinical trial design question, Jocelyne, and then maybe you can talk about how just that continuum that you've architected to take field learnings and bring it back even to the preclinical stage if needed. What I would say, Matthew, is we try to incorporate, in all humility, all of the best thinking around the world when we design a clinical trial. The current Uplift trial resulted from consultation with people who have experience using transcutaneous therapy. We also spoke with Praxis, and we did our best to get the sort of the voice of the community as part of that. And now, as we begin the process to shape the pivotal trial for our implantable device for blood pressure and trunk control, we're doing the same thing. So we're going out, and I flew back to the US during COVID last month and so on. But we're doing our best to reach out to the thought leaders in blood pressure in the spinal cord injury community and get their thoughts and convene them and help us shape the protocol. I mean, that's really what's critical. So we do have this deep relationship with NeuroRestore, but we're a global company, and we're doing everything we can to harvest voices and perspectives everywhere. So, I've learned a lot during this conference, and some new people that we want to speak to, and hopefully, I don't think that they were all aware of ONWARD in the past, but we want to build those bridges now.
Well, it was one of the questions I wanted to ask you, and you just started answering it, which is, how are you getting info from the community? People with spinal cord injury, what's that kind of relationship, and how does that roll up into your strategy now as a company going forward? And certainly appreciate you being here. This is a great opportunity to sort of educate people about who you are, what you're doing, where you are, and what they can expect. So you partially answered that question. What does that relationship look like going forward?
Yeah, I'm thrilled to answer that question. First of all, as an emerging company, we've tried to be involved. We were at this conference last year. We're here again. We were at Asia as well, and Gregor and Jocelyne delivered the keynote lecture there. So we're doing our best to be present. I think we're probably more present in Europe than the US, but that's changing very quickly. We also, though, really strongly value our relationships with the patient advocacy groups, whether that's Praxis or Wings for Life or Spinal Research in the UK or the Christopher and Dana Reeve Foundation. And in fact, the Reeve Foundation invested in our company. They're one of our shareholders, and so we leverage all of these relationships to get, in business it's called voice of the customer, but here I like to call it voice of the community. And we also have even forged some important consulting relationships of late, such as we work closely with Scott Chesney, for example. We want to really be part of and a critical family member of this community, so we know what's going on, and we can harvest learnings, but also the community knows what we're doing at the same time. For me, it would be I'm going to use the word tragedy. Let's say if somebody flew to Thailand to get an implant right now, knowing that we have purpose-built technology that, if everything goes well, is going to be FDA approved and is backed by programming and field support and all the things that one would want if a person would get an implant. So I think it's quite important that the community knows what we're doing and that we have the resources to make this happen.
Let me throw one, because you brought it up again. You're triggering all these things that are questions that I wanted to ask. One of the things that I, as an advocate, have some concern about is the clinical space, right? And I know that's got to be a piece of the puzzle. And there's another thing that Florian spoke about yesterday. Tell us a little bit about that development when you kind of laid out that picture of technical support and community buy-in. Clinician buy-in is huge, right? And in particular, and obviously I'm speaking mostly from the American healthcare model, which I'm not necessarily the greatest fan of in its current iteration. The clinical capacity, who is it that follows? Who is it that you're going to reach out to that follows a patient, say, who has an implant? The neurosurgeon is not going to be the one to follow them. Is it the company that's going to work very closely with the clinician who's following that physiatrist or urology? Or, there's kind of it's sort of fuzzy as to who's the clinical expert that takes care of neuromodulation in the real world clinical space.
Well, there are a lot of stakeholders here, and there is some complexity, but at its core, it's a relatively simple, again, using business speak, go to market. Okay. And that so many of these people with spinal cord injury are cared for in these specialty rehabilitation clinics. And so the real decision maker is and they have different names depending on the place in the world, the neurologist, the physiatrist, whatever, the rehab physician. They're the ones who will determine whether a person would benefit from the transcutaneous or the implantable. And if they benefit from the implantable, again, it's relatively simple. There are functional neurosurgeons like Jocelyn. This is a subset of neurosurgery who deal primarily with device therapy, DBS, and spinal cord stimulation. And they're the ones who would do the implant, a very similar implant to that which is done for pain. And we just need to support those implants with our field engineers to make sure the lead's placed in the right position and that it's programmed properly. Then our view is that those people would return to those spinal cord injury clinics for ongoing care, and we would support them with periodic checks. This is actually quite normal and routine. You see it with pacemakers, implantable defibrillators. It's been done this way in cardiology for decades.
Yeah, it's funny that you mentioned that, because the image I have in my mind is, back in my days when I was managing an ER, someone comes in to the stabilization room because their pacemaker is throwing them into an arrhythmia, or it didn't kick in.
2 hours.
So back in the old time, you'd have to call the Medtronic guy.
Yeah,
right?
He'd come with his magnet, his donut, and
his magnets, right? And so that's the image I have in my mind. Obviously, that's maybe a decade or decade and a half old, but that is part of my question in terms of what is the support that ONWARD provides post and with whom, between
and that's part of what we have to build, and that's that we're going to use the capital from our recent raise last week to build that field organization. But that's envisioned at the right time, prior to the introduction of our implantable system, when we expect that to be approved, which currently we expect in late 2024. So our timeline would be to, again, if things continue to proceed according to plan, have our transcutaneous device, ARC-EX, FDA approved in early 2023, the implantable device for blood pressure and trunk control in the second half of 2024, and then something for walking and standing in 2025. And we'll build up our field organization, sort of in a stage appropriate way. Some of them will be in place to support the clinical trial sites, and then we'll add to that when the therapies are approved and scaled.
Great. I just wanted, for a bit of context, both for the audience and you you haven't been here through the 3 days, but there's been a lot of contextualization. And so that these questions are coming from a lot of that, right? And there's a lot of complexity to, taking a device from understanding a proof of concept all the way through to become a standard of care. And so I appreciate you being willing to engage with these questions and help explain them. And then lastly, I do want to say we do have to wrap up, but you mentioned it a couple of times, but I wanted to bring up first that you've had a pretty busy week this week and you've been pretty successful in terms of raising some dollars to invest in this. I think that's exciting. Care to comment on why you're so tired, maybe?
It's been a pretty challenging several months, actually, but I'm not allowed to talk too much about it because we're in a quiet period. But we had an IPO this week. It was actually we believe the largest medtech IPO in European history. We raised over $100 million to help build ONWARD, to build this company into a significant and meaningful part of the SCI community here. And we're going to use those funds to continue to invest in R&D, to conduct these multiple clinical trials that we have planned, starting with Uplift, and then to spool up to commercialize and scale these therapies and then support them thereafter. So I think that's really what's been missing in this field, is there just has not been a well-funded company with the requisite expertise and experience to fill in this translation gap. And we feel so fortunate and grateful to have raised these funds. It's not easy. The IPO market is changed quite a bit in the last month, but people believe in our mission. They see that this is a big unmet need and they want to support it. And they showed that this week.
Excellent. Well, congratulations. Congratulations to all of you. Very much appreciate your time being with us. Gregoire, Jocelyn, your research and background, clinical work, also appreciate that. I do want to extend an offer to you. When you go back on the live stream, there's a whole bunch of questions that I didn't get to, comments that I didn't get to. There's a lot of back and forthing between people there. If you're willing to show up there and answer some that you're able to, obviously, there are limits to that. And then also, we will, at the close of the virtual symposium, we are inviting audience members to an open forum, breakout Zoom calls on each of the topics from today, and then kind of an open forum to discuss. We welcome you to show up there, and you can answer questions directly from folks if you have the time and are able. I know you're way ahead of the day from us, and Gregoire is in his vampire lair there at the chalet. His face. Really appreciate you and your time.
I would have loved to come in presence and tell you all about what we are doing, eyes in the eyes. Next year.
I wish we could have done this in person very much. I think we all share that. Thank you so much.
Thank you.
Thank you very much. Onward.
So that's it, folks. I just want to wrap up with a few comments. I know once again, we went over a few minutes. Thank you for your patience. I want to extend a thank you to all of our presenters, all our panel participants, the advocacy spotlights, as well as the contextualizing stuff. Really appreciate the commitment of time and the passion with which everybody brings to these issues, these puzzles, these questions, and this work that we have ahead of us. Thank you for being here with us. Again, extend a thank you to our sponsors for allowing this whole program to go on. A couple last things. Again, I think what we've tried to communicate throughout these 3 days in particular is really try to impress upon everyone the importance of the relationship between the spinal cord injury community, the people who live in closest proximity to the problem of paralysis, with all the other stakeholders engaged in the process. And it's incumbent upon us in the community to become more savvy, more shrewd, pay close attention to what's going on and who's doing what. And so I hope we were able to impress that upon you. We will continue our efforts at U2FP to do that in any of the number of ways that we do, through our podcast, the legislative work, this ongoing symposium, some of our blogging and writing about science. So again, I hope that came through very much, because back to that first day comment, I really believe that it's together that we define a win. I do not think it can happen separately. We are the community of people with spinal cord injury are the target, and the win has to come through this community. So again, thank you for your time. Thank you for being here with us. Thank you for your participation. And please, if you're able, join the breakout Zooms on each of these topics. And we'd love to see you there and continue the conversation. Oh, one other thing. Please fill out the conference survey. We very much need and want to hear from you, so please do that. Thank you. We'll see you again.